Good morning or good afternoon or good evening to everybody. It's my pleasure to welcome you to the NHGRI Machine Learning in Genomics workshop. My name is Mark Craven. I'm on the faculty at the University of Wisconsin, and I serve on the NHGRI Data Science Working Group and the NHGRI Council. And it is my honor to be co-chair of this workshop along with Trey Eidecker, although really it's the NHGRI staff who've done all the hard work in organizing the workshop. So we have a truly stellar lineup of speakers and session moderators who are working at the forefront of machine learning and genomics. And in just a minute, we'll get to the program. But before we do that, I'd like to introduce and turn the floor over to my co-chair, Trey Eidecker.
Hi, so as Mark said, I'm Trey Eidecker. I'm with Mark, the co-chair of this meeting. I am a professor in the Division of Genetics in the Department of Medicine at UC San Diego. You'll be hearing a lot more from both Mark and me as the meeting goes on in the various sessions and then in the session wrap-ups at the end of every day. So I think with that and without further ado, it's my great honor to introduce our Institute Director, Director of the National Human Genome Research Institute, Dr. Eric Green, who will give some welcoming remarks.
Well, thank you, Mark and Trey, for your introductions and actually for your remarkably valuable leadership and help in putting this workshop together. And I want to welcome everyone to day one of NHGRI's Machine Learning in Genomics workshop. Now, for those of you who may be a little less familiar with NHGRI, the Institute is one of the 27 institutes and centers that makes up the US National Institutes of Health. Of course, the NIH is the world's largest funder of biomedical research. Now, as an institute, we every once in a while put together and publish strategic visions that really help to guide the field of genomics. And our latest was published late last year in 2020. We released our new strategic vision, which we think really details the most compelling areas to pursue in human genomics in the coming decade. Now, the strategic vision is organized into four major areas: guiding principles and values for human genomics, sustaining and improving a robust foundation for genomics, breaking down barriers that impede progress in genomics, and finally, compelling genomics research projects in biomedicine. Now, needless to say, artificial intelligence and machine learning came up multiple times during our strategic planning process and really are identified in the strategic vision as areas that present tremendous opportunities for genomics. And by prioritizing novel statistical methods and considering aspects of machine learning that could be complementary to more traditional analyses of genomic data, it seems very likely that machine learning will become integral to the next breakthroughs that we expect in the coming decade in genomics. In another area of focus, though, it's important to understand that we need to understand how machine learning is going to be used and how it sort of fits into the context of important ethical, legal, and social implications of human genomics. And that will be another really important area to consider as we think about the productive use of genomics and the implementation of genomic medicine. If you want to read more about our strategic vision, it can be found on NHGRI's website, genome.gov. But it wasn't just NHGRI that does strategic planning in this area. At the NIH level, there's been increasing interest about artificial intelligence and machine learning. And back in March of 2019, the Artificial Intelligence Working Group that was established by the NIH director, Francis Collins, as part of his advisory committee to the director, they finalized their report, which detailed the potential role of artificial intelligence and machine learning in the future of biomedical research. And the report identified opportunities, challenges, and outcomes of implementing artificial intelligence and machine learning approaches across all the NIH institutes and centers and all of the areas of biomedicine that NIH is interested in. Now, the working group's final report concluded that the computational and biomedical communities are poised to jointly drive transformative progress in biomedical research, leading to new insights into how all living systems work and in care delivery, leading to improvements in the health of all humans and all communities. And a specific recommendation of the working group worth highlighting is recommendation number 8. The NIH should continue and expand support for engagement with wider artificial intelligence and machine learning communities. This approach was piloted at the NeuroIPS in December of 2019 and should be expanded to other conferences and other opportunities for convening experts from different fields. And thus, this makes a lot of sense relative to this workshop. And so at this week's workshop, we hope to hear from different members of the artificial intelligence and machine learning fields to get a more complete picture of the opportunities and challenges that will guide future NHGRI directions in our area of interest, of course, that being genomics. Now, related to the recommendations of the Artificial Intelligence Working Group of the NIH Directors Advisory Committee, the NIH recently issued important funding opportunity announcements for something known as Bridge to Artificial Intelligence or Bridge to AI program. And the FOAs are listed here. And if you want to read more about this, I would send you specifically to this website that's listed here on this slide, which I'm sure you could also find by Googling. So for this workshop, what is our intention? So in light of all these developments, both at the NHGRI level, at the NIH level, we have decided to spearhead efforts to push the boundaries of machine learning in genomics. Now, we understand the value of engaging the community for this, as we always do, which is the reason behind convening this workshop. And in fact, the initial feedback from the community was a guiding factor in shaping how we put this workshop together. Now, the major goals of the workshop are to stimulate discussion around the opportunities and obstacles underlying the application of machine learning methods to basic genomics and also genomic medicine. Also to define the key areas in genomics that would benefit from machine learning analyses, and also to identify and shape NHGRI's unique role at the convergence of genomic and machine learning research. The 4 sessions of the workshop will reflect broad topics of the machine learning field with ample time after presentations for questions and comments. And all the feedback from the question and answer sessions and post-meeting survey will be recorded and available for review subsequently. Now, workshops like this do not happen with a lot of work from a lot of people. So I want to thank, starting with the co-chairs, Mark and Trey, but also the other members of the Genomic Data Science Working Group of the National Advisory Council for Human Genome Research for their support and planning and also moderating this workshop. For those unfamiliar with this working group, it was created as a subcommittee of NHGRI's Advisory Council to provide ongoing guidance related to data science as it relates to genomics and NHGRI programs. I would, of course, also like to thank the NHGRI Organizing Committee, as well as 2 very critical components of the Institute, our Information Technology Branch and our Communications and Public Liaison Branch for their hard work in all of the IT, communication, and other logistical aspects of putting a workshop like this together. And finally, of course, I want to thank the speakers listed here for presenting their research throughout the workshop and making themselves available for questions and feedback from the audience. This could not come together without the generosity of your expertise and knowledge and willingness to join us throughout the next 2 days. And lastly, of course, I want to thank all of you for joining us today and tomorrow. The interest in this topic turned out to be, well, probably the best way to describe it would be huge. I can tell you that as of Friday, there were over 3,400 people from 73 countries who registered for this event. Overall, this workshop then promises to be highly influential in guiding the field of machine learning and in genomics. And I really do look forward to the next 2 days as we see this unfold. And so with that in mind, I'm going to hand the session and the screen over to Dr. Shannon McWeeny, who's going to start the keynote session. Thank you very much for joining us, and I look forward to spending time with you over the next 2 days.
Hi, I'm Shannon McWeeny. I am a member of the Genomic Data Science Working Group. I'm also a professor and division head at Oregon Health and Science University in Portland, Oregon. And it is my honor to be the moderator today for the keynote session. Starting off, we have Eric Topol. He is the director and founder of the Scripps Research Translational Institute and the Gary and Mary West Endowed Chair of Innovative Medicine at Scripps Research.
Machine Learning and Genomics workshop. I'm pleased to join and discuss how genomics is really moving forward with AI. First, to point out, of course, that as you know, we've hit the 20-year mark for human genome sequencing. And what that's done, this incredible milestone, has led to extraordinary amounts of data. And this is really interesting because we've gone past yottabytes. And the question is how many data sets that are going to start to exceed levels that we had never anticipated, with genomics being a part of that. So it's been postulated that we should start renaming a new unit of bytes to exceed the level of data that we have today. So how are we going to deal with all this massive data, some of which, of course, is through not just DNA sequences, but all the different layers of biology? And that makes us turn to neural networks. And so the neural networks, these deep neural networks, although they've been compared with the brain, they really are not all that similar. They're basically using layers, really hidden layers of artificial neurons to process the data of inputs, which can be of massive scale, and to come out with a proportional level of layers to get outputs of interpretation. And genomics is just one of the many areas in life science that's going through an AI revolution. And that's, of course, the focus of the workshop. But also to give a point, this is very early in time point. It's only in 2015, not even 6 years ago, when we first started to see these convolutional networks for genomics like DeepC and DeepBind. And already now, we've seen just in recent days, the emergence of a dedicated institute at the Broad Institute, the Schmidt Center, which has a bunch of partners as shown here. And it's basically taking into account that this has now been a field that's developed where the data is massive and we need AI tools to process it and extract all the valuable information and knowledge. So first, let me point out that most of AI in medicine so far has been with images, that is like this chest x-ray, a scan. And that's a whole lot easier for AI interpretation than a DNA sequence. The reason being is that the pixels in a scan have only limited interaction with their neighboring pixels, whereas in a genome, there is 3D relationships that can be long range. And so the ability to interpret a genome accurately is a far more challenging task. But we have seen imaging being used for AI in genomics. And a great example of that is this used in children. This is the ability of Deep Gestalt, a deep learning algorithm developed in Israel, whereby a smartphone picture, which has been trained in hundreds of thousands of images, to accurately make the diagnosis, at least tentative diagnosis, of the chromosomal genetic abnormality. It has over 90% accuracy, and the number of syndromes it can yield accurate diagnosis keeps increasing. So we have seen some image interpretation used in genetics and genomics. But what we're really talking about today is the actual data, biologic data, whether it be DNA or RNA or methylation or 3D, transcription, binding sites, all these things, and applied in specific instances like tumor or cancer, being able to distinguish the regulatory genome, the functional genomics, have been approaches of deep learning in genomics today. And there are generally 4 different classes of neural networks. The most straightforward one is just classification, fully connected or feed forward. Most of what we're talking about is convolutional for DNA sequence, but some using time or recurrent neural networks. And then there are graph convolutional. So these different neural networks that are used in genomics are example here. This is to determine a single transcription binding factor, but it can be multitasked for 2 transcription factors, and then it can be integrated with DNA sequence and chromatin accessibility. And this is a really good review article that, even though it's almost 2 years old, in Nature Review Genetics, I certainly would recommend it to you. And in that same review, there's the idea of haplotypes, and this extends that. This is just the work done in just a couple of weeks ago, whereby a haplotype for variant calling was using a Bayesian model. This is so-called octopus, getting much higher sensitivity and specificity than prior variant callers. And this is one of the big issues when we have genomic data, is being able to call what is a variant and what isn't. And this is the output of octopus compared to its predecessors like deep variant or GATK and others. And consistently, octopus outperformed. So that's just to show you how we continue to eke out improvements, iterative improvements with these different deep learning algorithmic efforts. Now, another part of DNA sequencing is the ability to predict these variants. And here you see AMBER and how it predicts better than the original one, Deep C and Deep Bind, and the heritability enrichment. So this again is another recent paper, again exemplifying this staged improvement and able to predict variants. There's also many other aspects that we see continued jumps in the use of deep learning. Here is an example of differential gene expression, and this was seen across all the different tissues in the body. But we also have seen it for predicting transcription factor binding, and this is agent bind. A lot of these algorithms have deep as their first part of their name, but here we're starting to see a little more creative names like agent bind. And this is for splice prediction from the sequence through a deep learning algorithm published a couple of years back in Cell. There's also the ability to distinguish rare variants for undiagnosed diseases that was published last year in Genetics in Medicine. Now, one of the areas where genomics is becoming more used in the medical space is in cancer and understanding, deconstructing the genomics of a person's cancer. And what's striking here is more reliance on the liquid biopsy, that is cell-free tumor DNA, plasma DNA that is from tumor, which now through deep learning algorithms can be analyzed to determine what is the primary source of the cancer, which previously it was yes, no, there's cancer. Now the ability to take this approach to understand its source. And that's a step in the right direction. And we're increasingly seeing liquid biopsy used in cancer. This is a welcome sign of progress. Also, the ability to pick up the mutations. This is an example in prostate cancer. Here, the standard method on one side, and then on the other is the deep learning and picking up far more variants, as you see compared in the deep learning. So our ability to extract the relevant information through deep learning in cancer has certainly been enhanced. But also, unexpectedly, our potential ability to predict the evolution of the cancer. So when you know a particular variants that are present in a tumor sample, you could actually predict when and what variants will be appearing over time. And this is just the beginning of that work. It's an exciting direction. Unfortunately, like so many other things that happen in the reports, it gets somewhat exaggerated. And here is the UK coverage of that robot war on cancer. AI predicts tumor growth. Well, that's of course a little bit of hyperbole. Now, on the cover of the New York Times Magazine at the end of March was a SARS-CoV-2 virus sequence, which has about 30,000 bases. And it's bringing it into the mainstream. And we haven't paid enough attention to pathogen sequencing. We've been thinking and talking so far about whole human genome sequencing. But what's interesting is, again, just like that prediction of cancer, where it's headed in a particular patient, the idea that we could predict a strain of influenza? Or where is SARS-CoV-2 going? By learning the language of the virus, and this was published earlier this year in Science, for influenza being the rich data set we have, and one that's developing right now, of course, is for COVID. Now, another tool that we have, before getting into some examples of how they can be used, is transfer learning or meta learning. That is, taking, in this New Yorker article, a very interesting piece that the use of a deep neural network for interpreting what type of pastry that ultimately was used in transfer learning to understand cancer. And also we have seen this meta learning, that is learning from learning, being applied to genomics like single cell sequencing or adult cancer genome atlas, now to pediatric genomics. And this is just an example of transfer learning in single cell RNA-seq, which are big data sets that can be hard to interpret, and putting them through a transfer learning approach. Now, the idea also, of course, is that we could analyze samples from large numbers of individuals and integrate that in the immunome. And here is from 11 million T cells from 40 patients, controlling for things like batch effects and sample preps. And this is yet another example of what you can do with a deep multitasking neural network. And of course, there's ability to analyze gene regulation and understand epigenetic regulation, as was published just in March in Nature Computational Science. Now, CRISPR obviously is the biggest breakthrough in life science of our era, of our time. And the idea that we could use AI to guide CRISPR is now been firmed up through many different studies. This was from the Microsoft group they're using AI to predict off-target effects of CRISPR guide RNAs. And then there's a series of articles that I've just highlighted here of AI CRISPR editing. Here you see whether it's in T cells or whether it's with deep learning with network-based gene features for guiding RNA for gene therapy. So not only has AI helped with CRISPR predicting off-site and predicting the right guides, but also in coming up with the best approach for gene therapy. I just want to point out that sequencing is getting more complex. Now we're seeing in situ sequencing, which is going to increasingly generate massive data sets. Again, why we need deep learning tools to interpret these data. And just for those who are intimidated by equations, this is an article that talks about multi-omics. And this is a problem we have, is integrating multiple layers of different omics, like the genome sequence, single cell RNA, epigenome, proteome, and these different layers. And this is a complex task that is yet to be fully actualized. Now, one type of AI, nearest neighbor analysis, deserves mention, because this is the idea that we could have digital twins from a massive resource infrastructure. And this is something that is really coming into play now, because of Tempus, which is a company based in Chicago that's largely been cancer dedicated. I'm an advisor for that company. It has generated a massive amount of data in cancer, now with 200,000 patients sequencing tumor and multi-omic data, along with electronic health record, treatment outcomes, pathology, and scans, relevant scans were digitized, and even more, they don't have full data sets. And the idea here is that we could do nearest neighbor analysis and find individuals at the time of one person being diagnosed, matched as closely as possible to then be able to predict treatment and outcomes. So this is, instead of clinical trials, which often have only a small percent of patients that are relevant for the patient in presenting in question, now we would have very precise potential matching. This has to be validated. It has not yet been to date, but it's a rich resource, and cancer is just the beginning of where this can go. Now I want to go through two projects to close that are real world projects that exemplify some of the issues. One at Scripps Research is our quest to take sparse data, namely the data that is found in arrays such as Affymetrix or Axiom with 4 to 5 million variants, and to then be able to predict the 80 million genetic variants. And so this is a task that has not yet been accomplished that we've taken on, and it's a very challenging one. And Raquel Diaz in our group is a KL2 scholar in our CTSA program, and along with Ali Turkamani has been leading this effort, starting on 9p21, which is the region of the genome, which is extensively regulatory enhancer rich. So it's a very complex area. Then currently moving on to chromosome 22, which has about 1,000 very complex regions, and not yet, and you'll see why, to the level of the whole genome. And the whole idea is using unsupervised learning. So rather than the types of AI like hidden Markov model or Bayesian, or even just regular supervised learning with deep neural networks, the idea is to use an autoencoder, which works through a bottleneck to get data out, and also GANs, or generative adversarial networks, that were really originated with Ian Goodfellow, here being shown about real or synthetic data from genomics. Well, we have taken the approach of using an autoencoder, but not just any autoencoder, what's called a denoising autoencoder for this imputation project at Scripps. And so what that denoising accounts for is the ability to deal with missing data. That's where the X's show on the input. And everything is about the inputs if you're going to go through this autoencoder and impute the right variants at a much level of granularity or depth. So here is comparing what has been the accepted standard, which uses a hidden Markov model, so-called Minimac, compare with our denoising autoencoder. And what you can see, surprisingly, it didn't do very well. It underperformed. And in fact, it was something like 79% versus 21% for having the correlation. And so we said, well, why is that happening? Why is our, what should have been this great deep neural network denoising autoencoder not working as well as a more primitive form of AI? And what it turned out was all related to the inputs, as you might have suspected. And when we adjusted the inputs using 30,000 virtual babies, the synthetic data with far better admixture, that is, ability to generate a much bigger data set with much more diversity, well, then we had this genomic data augmentation gave us the ability to simulate the Minimac. And that is important because an autoencoder like this can move at a much faster speed, much less computing time, and it can be continued to be tweaked to be far better. So this then gives us a double benefit, not just to deal with the imputation, but also to then apply that to things like polygenic risk prediction and multitask deep learning, which is the end goal of this initiative. And you can see here the advantage of the autoencoder that we use versus Minimac, the runtime and the accuracy, while getting equivalent accuracy, how much the time reduced to process this data. So it's really an exciting advance because the more genomic data we have, the more we can use these unsupervised learning tools of deep neural networks. But to point out, this is a graphic processing unit GPU guzzler. We're using 4 models per GPU, running 18 GPUs for this project of chromosome 22. Each model takes 3 to 5 days to train. And to extrapolate that for a whole genome, we'd need 500,000 GPUs. So that's why we haven't gotten there yet. We're just getting chromosome 22 nailed down. So the point being here is to get not for the end user once the validation is done. But to get this imputation work accomplished, it takes a lot of computing power. And this is what the A100 current state-of-the-art GPU from NVIDIA looks like. And this is how much it costs. These are the specs. And this is over $13,000. So putting a whole bunch of those in your cart would really add up very quickly, especially if you're trying to do a whole genome. Now, that's one project. The other one I wanted to highlight is with our partner, Rady Children's Hospital, the largest and really the children's hospital, pediatric center of San Diego County, which has 3.5 million people centralized in this one facility. And Stephen Kingsmore and his group, working with us in our CTSA, has developed the program in the world for rapid genomic interpretation, acquisition and interpretation and management change for sick neonates, not just newborns, but also even in children. So this typically is in a neonate. And what's fascinating here is, of course, every minute counts, because if a diagnosis isn't made quickly, it could lead to brain damage. It could lead to the death of the newborn. So that's why time is essential. And now this group has been able to take from sample from a critically ill infant to management of that infant in 13.5 hours. And this is using multiple different AI tools. And it, to me, is the best example of AI in medicine today, which is interesting because it upends the usual story of starting in adults and only eventually getting into pediatrics. This is just the opposite. So the steps are, number one, taking the structured and unstructured data from the medical record, from the electronic health record. That takes less than a minute, actually 20 seconds. And then it's the variant calling, which is happening concurrently. That takes a bunch of hours, and the automated diagnosis, and eventually the automated management. And this all can be done in a time frame of just over 13 hours. So just to take you through the steps, Rady is using CliniThink or CLAMP more recently to do this clinical natural language processing, which is working also at the unstructured level of data in the electronic health record to pick up all the relevant terms. Then there's the automated interpretation, and this is using two different tools, Fabric Genomics, GEM, and Invitae's Moon, which prioritize and ranks the variants and gives a score. As it gets that score over 1.5, it gets to very high likelihood of being an accurate diagnosis. And then the third step is this genome to treatment management. This is a home-baked effort at Rady that incorporates multiple different AI algorithms, Alexion, which has the data from all the literature, as well as Rancho Biosciences, taking the compilation of all the resources and then coming up with a management for the neonatologist or pediatrician on how to manage the condition that's been diagnosed. This is really extraordinary, a whole bunch of different deep neural networks to get the answer to manage a sick neonate. And that takes me to the end, which is we think in genomics too much genome centric. We want to see as much integration with other layers of biologic data, the electronic health record, the environment as possible. And so eventually we will get to this point with genomics being a fundamental layer of a multidimensional effort of real-time processing of a person's data, be it for a virtual medical assistant or for a clinician caring for a patient. We aren't there. Deep learning is not enough. Hybrid models will be required, but eventually this is something that is an exciting frontier in the years ahead. So with that, let me thank all my colleagues at our institute, SRTI, within Scripps Research, with whom I have the real pleasure to work with on a day-to-day basis, and all the different funding support that we have from NIH, from NHGRI, and from all of us. And I look forward very much to your questions and our interaction. Thank you.
Thank you so much. That was fantastic. Our next, just as a reminder, we will be taking the questions at the end for both of our keynote speakers. Our next keynote is Bradley Malin, who is the Accenture Professor of Biomedical Informatics, Biostatistics, and Computer Science, as well as the Director of the Health Information Privacy Laboratory at Vanderbilt University.
Good morning. I'm Brad Malin from Vanderbilt University and Vanderbilt University Medical Center, and I'm speaking with you this morning about various challenges and opportunities for machine learning in genomics. So let's jump in. One of the first things that we need to talk about is that it's a process. The entire learning process is facilitated by the data that we collect. And this data comes from a variety of institutions, individuals, and organizations all around the world. And once you collect all the data, we're going to be pushing it through some type of a magic box. And I don't think that everybody completely understands all of the frameworks that machine learning facilitates. But at the end of the day, what it's doing is learning over the information to try to generate a model and give you a relationship between all of the different facets of individuals that have been fed into the system, whether it be about the genes, whether it be about the single nucleotide polymorphisms, or just general variants. However, the process is not one way. Once you get a model, you need to go back and figure out, was this actually a good model? Did the system learn in a manner that provided me with intuition that supported the biology that we're aware of already, or provided intuition that gave us new direction to proceed with that we weren't aware of before? But it still makes sense. Now, if it doesn't, we go back, we check to see if the model is any better. However, it may also be the situation where we don't have the right data. And if you don't have the right data, now you need to go off and augment the system as it's been designed. And what that means is trying to collect additional variables about individuals, but also possibly about trying to collect new data about people that you haven't seen yet. So there's 4 different topics that I want to cover today. We're going to dig into them, but I'll leave a lot of time at the end so that we can have more of a discussion during the Q&A. So the first topic that I want to discuss is that bigger is not always better, but it can be. So I call this the safety in numbers problem. First, I think everybody recognizes the process that I just explained a moment ago. But when you have a single site that's actually performing machine learning, at the end of the day, they want to know, did what I see actually make sense at other institutions? So it makes sense for an institution to say, hey, what is everybody else seeing? And it might be that I don't have sufficient information in order to verify that what I saw was statistically significant. So I want to go check and see what others have. So there's a number of other organizations that are going to be collecting information. And we realize that there's a lot of barriers to sharing this information, but we have various networks that have been established to facilitate this process. Now, when you do this and you check to see if what you have replicates in other institutions or with other people's data, then this is about robustness. This is about ensuring that everything that you have is either replicable or generalizable to a certain degree. Now, this is not easy to accomplish. We've had several consortia that have pulled this off, but we're moving in this direction. But to really do this in a meaningful way, we're not going to have to just replicate what we've come across. We need to broaden the data. And what this entails is typically we're dealing with the DNA of an individual. And everybody's recognized that there are other resources that need to be brought to the table in order to facilitate deeper investigations. Now, this could include the socioeconomic status of the individuals to whom the data corresponds. It could be information that comes out of their medical records. And for the last 15 years, we've been doing this within the Electronic Medical Records and Genomics network that NHGRI has been sponsoring. But it also might be about taking clinical trials that have been performed and expanding them with real-world evidence. Regardless, this is about linking all of this together. Now, these are only a couple of resources that I've shown you. There are a number of others that could vary in the amount of information that we have and the comfort level that people have in providing them. And these are the non-traditional pieces of information, everything that includes an individual's over-the-counter or retail purchases, possibly at a pharmacy or at a grocery store, what they're doing in a social environment, whether that be through social media or just general social interactions in terms of who they're hanging out with or where people are receiving influence from. It could be about how much energy they have or what they're doing on a daily basis in terms of moving their body around. That could come from fitness records, such as an Apple Watch or a Fitbit. And then it could also be about lifestyle decisions, things that we ask people or just generally observe, such as how much alcohol do they consume, do they smoke, in general. Now, this is one little piece of the puzzle still, because this is about what one person who might be a parent, for instance, has actually done and has been documented. But we don't just want to look at one person. We want to look at the relationship between individuals. And so we want to look at either mom-baby pairs or father-child pairs, and not just a single child. We want the entire genealogy or the pedigree, and then track and integrate all this information to facilitate the type of investigation that is not just a one-off. Now, I think everybody recognizes that over the past several years, it's become clear that the going broader perspective is not really sufficient to provide society with the type of insight that we need when we're performing any type of learning with this type of data. And the notion of bias has crept in. Now, what exactly does this mean? This is an example of information that Steph Devaney from the All of Us program put in a paper a couple years ago. And what you can see here on the left is the number of people who fit a certain racial profile that have had their information used in gene association studies. And what you can see is that it's on the order of close to 80% where it's Caucasian background, European ancestry that have had their information used. Now, it's a little better in terms of the bias when you move over towards what studies have been done. So as you can see, even though we have 80% approximately of white individuals in the resource or resources, that the total number of studies in which they've been used is only around 50%. Now, this doesn't necessarily mean that these studies are all equal. This is just a count of the number of studies that have been performed, and some of them are of a larger scale than others. Regardless, it's still clear that there's a bias with respect to when information is being used and studied. Now, the implications for this have been non-trivial, because when you learn models, such as in this situation, there were models that were learned for 17 qualitative trait loci. When you make these types of, when you make predictions based on this information, it's pretty clear that the generalizability of the models really falls to the wayside as you shift away from the population from which the model was based. And so if the model was based on European individuals and the predictions are made on European individuals, then the variance associated with its predictive capability remains pretty small. And so the point estimate that you had in the initial studies holds true. However, as you start shifting towards other populations, such as going towards Southeast Asians, East Asians, and African populations in particular, or populations of African American heritage, the predictive capability drops to a point where it could be as good as what you saw, but it could be almost 75% worse than what you expect it to be. It's a known phenomenon. It's just that this happens in genomics at a scale that we really haven't seen, simply because of the number of variables that come into these models. This was known, for instance, at least 30 years ago, when the Framingham heart study data was used for facilitation of the design of heart attack risk or cardiovascular risk. But we're now seeing this on an even larger scale. So there has been some movement to try and change the situation. And I'll highlight that in the All of Us program, which I have a relationship with, this was a notion that was really taken to heart from the beginning of the study. And it's about really enhancing the resources that we have to not just include people who have typically been involved within biomedical research, but broadening this towards underrepresented groups. As of about a year ago, with the 250,000 people whose specimens and data that we had collected, we had deliberately selected for populations that were not just white in nature. And so over 20% of the population that we had established or collected information on was of Black or African American descent, and around 17% were of Hispanic background. Now, this doesn't necessarily mean that we have completely changed the problem or fixed the problem. What I'm showing is that you need to be deliberate about changing your perspective and creating data sets that are going to lead to greater equity in the research enterprise. At the same time, I said bigger is better, and you probably were thinking about data. But in reality, it's the entire process, and that process includes the people who are performing the investigations. Because when you're going about learning models and just performing learning in general, people make conscious decisions about what models need to be used, as well as what is the pre-existing knowledge that we bring to the table. And that knowledge is dependent upon the people who are actually performing the investigations. So you need to broaden the population that are actually performing these investigations. Now, this has been recognized by NHGRI and the NIH in general, but simply broadening the set of investigators who are at the table is one thing. You need to be able to broaden that population with a skill set that can actually do the types of analytics that we're talking about. Otherwise, you'll have scientists that are sitting at the table saying, you know, we should do studies in X, but they really wouldn't understand how exactly you do that study. Okay, so this is the first challenge that I believe is going to help drive where we go over the next 5 to 10 years. The second one I want to talk about is about cost effectiveness when we're performing machine learning. One thing that I hear over and over again is that we're going to move to the cloud, and we've been moving to the cloud for at least 5 to 10 years. And the cloud is wonderful. It provides dynamic compute and elastic compute capabilities, but at the same time, there's a cost associated with this. And I'm not talking about hidden costs. I'm talking about explicit, you put your money on the table type of costs. So the process that I described below earlier was really just a small summary of what's going on with the scientific enterprise, where you take data forward, you push it into your machine learning framework, whatever it may be. But then in the cloud environment, you're going to pay for every analysis that you're going to run. So you pay a little bit, or you give a little bit of money to your graduate students and they pay a little bit and they run their study, and out comes junk, absolute junk. This is fully expected the first time around. You either didn't tune your parameters correctly or the data wasn't loaded correctly. Whatever it was, the money is spent and junk has been generated. So what do? Next step, you wipe that data, you give your graduate student another $100 and they run the study again. And out comes, it's something that's a little better. You know, it's still a little junky, and you look at it and you say, well, you know, it's heading in the right direction, but let's rerun the study again. And you do this over and over and over. And this is a normal scientific process, but unfortunately, in the context of the cloud, you're in a situation where you are constantly paying for compute. And so after you've done this on the order of 1,000 times and spend $1 million, then you get to the point where you go, aha, I think I've got an interesting paper. This is not effective. This is not scalable. This is not supportable. In order to make this better, we need to create algorithms that make sense of data with a smaller model, or figure out how to more cost effectively run lots of models simultaneously so that we can sift through how we're actually generating the data, how we're actually generating our findings, to direct workflows in a manner that makes it much less likely that we're going to spend our entire budget simply to try and sift through how best to use the online cloud computing environments that have been established. So the third point that I'd like to talk about today has to do with data sharing and making data more widely accessible. Now, before I do, I'd like to begin with a cautionary tale. And in this tale, we are in an environment where people have begun sharing their genomic information into the public setting. And this is an artifact of the direct-to-consumer genomics revolution. One of the ways in which people have decided to take genomics into their own hand to facilitate learning is making their data accessible in websites where they get to discover relationships with other kin. Now, this sounds like a great idea. It facilitates discovery, but at the same time, it can lead to unintended consequences. One of those consequences is typified by the way that law enforcement has begun to use these resources as well. And the example that I offer to you is the case of Joseph DeAngelo, who is the Golden State Killer. The long story short is that this was a serial killer who in the 1970s committed a number of crimes in California. And when the case went cold, they lost the ability to discover him. However, the FBI did have DNA from the scene. And at a future point in time, actually several years ago, they were able to go to sites like GED Match, where they could put his genomic record into there and then discover who his relatives were. And they were able to find his third or fourth cousin and then build a pedigree and figure out, where is the individual that does not show up in the modern day society. And eventually the family members led them back to this individual who was in the state of California. And once they took his DNA, they were able to figure out that this is the individual that they've been looking for. This sounds like a great opportunity for law enforcement, and yet it turns into a bit of a concern for the rest of society. GED Match is not just a single one-off. Turns out that this type of a problem for Caucasians in the United States leads to information of a third or fourth cousin on around somewhere between 50 to 80% of the population. So if we have concerns with making information public, then one of the ways in which we might be able to solve this problem is to make the information accessible to only the algorithms themselves and learn over data behind closed doors. And so one of the ways in which this can be done is through the notion of secure multi-party computation. And in SMC, what happens is that you encrypt all the information and then you compute over the information to generate aggregated results without ever revealing what any particular record corresponded to. This sounds like a great idea, except in order to make this really work in practice, there's several things that we need to do. First, we need to have software that facilitates the rapid reconfiguration of any computable model so that we can evolve it with respect to statistical techniques as they change. Secondly, we need to be able to take advantage of the fact that some of these computations are best done in a software system, some of them are best done on hardware. However, it's probably going to be a combination of the two, and figuring out how to optimize that is going to require further investigation. Third, people are going to be using data, but they're not necessarily going to be telling you what they're actually using it for. And so this creates concerns about accountability. Now, one of the ways in which we might be able to address the accountability problem is through distributed ledgers. I'm not necessarily a large advocate for blockchain technology, but it might actually serve as the basis of what we're looking to support in the future. And finally, if we're going to create an environment where we allow people to compute over data that they can't see, we need to make sure that they're comfortable with that. A lot of scientists like to be able to scratch and sniff and make sure that the data is what they think it is. But once you take that out of their hands, there's going to be lots of questions over how trustworthy is this setting. So another way in which information is going to be, or there are expectations that information can be shared, is that we move from sharing real data to providing synthetic data out into the world. And instead of allowing people to perform direct hypothesis tests, we allow them to perform hypothesis generation to determine if it's even worthy of moving forward with deeper analyses. Now, we've been conducting research on the notion of synthetic data for a couple of years now, where we really cut our teeth on using deep learning frameworks to simulate electronic medical records data. And the framework associated with this is through what you call adversarial learning, but the details of this I'll leave to another time. What I will illustrate is that it does have the ability to scale up towards high dimensional environments. And what you're seeing here is an illustration of the correlation of the rates at which diagnosis codes for individuals show up in two random samples of electronic medical records. And what you're seeing here on the y-axis is the rate at which this information shows up in the synthetic data that we end up generating. You can see visually that these systems are not exactly the same, but they're moving in the right direction. In terms of translating this into genomic data, fortunately, just about a month ago, there was research to illustrate that it appears to be a feasible application as well. But we're really at the beginning of the innovation curve. So the last point that I want to bring up has to do with the movement of machine learning from research into its application and the notion of decision support. We're moving into a world of genomic medicine. But in order to do that, we're going to need to take into account the fact that there are hundreds to thousands of variables that are being used to create these technologies. The Food and Drug Administration has recognized that these types of machine learning or artificial intelligence-driven technologies are going to be useful. And they've already provided approval for over 100 different technologies. And this has really picked up over the last 2 years. But if you're going to go in this direction, several things you need to keep in mind. As I already alluded to, these are going to be large frameworks. There's going to be thousands of variables and lots of things can go wrong. We need that system to be verifiable. We need to know that the right model was used at the right time and who used it. And so in that respect, they must be auditable. We know that these systems are going to evolve over time, so we need to know which one was used when. And they must be explainable. We need to have the ability to tell people that this is the reason why this technology was used. And building trust is going to require having the ability to relate what the technology is doing into some understanding of the real world. And as I alluded to, these are going to need to be equitable. So several parting thoughts. One, we do need big data, but this data cannot just be deep. It needs to be diverse. Secondly, we need systems to be cost effective. We can't be spending millions of dollars just for computation. Third, we need to have trust in this environment. We need people to feel comfortable with just providing the learning or the results of the learning over the data instead of actually seeing the data itself. And then finally, we need to have explainability. We need to ensure that the way in which our system is making recommendations for action are actually in association with how the world works. So I thank NHGRI for their invitation. I thank you for listening to this presentation. And I thank the All of Us program and the Electronic Medical Records and Genomics Network for providing proving ground for some of these technologies as we begin to move forward.
So we're going to begin the Q&A. You have the ability to ask questions of either of the keynote or for both of the keynote speakers using a Q&A polling session. Only the Zoom participants can do this. And we already have a couple of questions that have come in. The first one is for Dr. Topol. This was about the really beautiful example you showed at the end, the collaboration between Scripps and Rady Children's. And the question was, how can that be distributed to other cities or centers? The person who's asking this had a child who was offered this at the NICU in another hospital, but the experience was that they'd have to wait. There would have been a long delay and they had to make a decision right away. And so getting samples from Spokane to Rady would have been a bottleneck in that case. Are there any thoughts or ideas on solving the challenge of getting this distributed to other centers? And then what are the challenges in broadly distributing a pipeline like this one?
Well, thanks, Shannon. It's great to be with you and Brad on this session. I do think the example with Rady is perhaps the most advanced use of AI in medicine today, and it upends the model where everything starts with adults and moves into kids. This is just the opposite. Now, what Stephen Kingsmore and the group is doing is trying to spread that throughout the country. So there are now multiple sites that are basically using the same tools, the same kind of flow of how to extract the data and get to the management side. So the hope is in the short term, all these refinements that are, it's constantly being tweaked will be universally available, not just in the US, but broadly. It's interesting because, just a couple of years ago, this was more than a 24, 36-hour story. And it wasn't to management, it was just to make a diagnosis. It's just getting so much better. But as the questioner brings up, diffusion of this is equally as important. We've seen enough validation that it really helps these sick babies and children. Now we've got to get it to become the standard of care.
Fantastic, thank you. The next question is for Dr. Mallon. This is in regards to cloud computing. And one of the questions is around trust. So this is coming from the perspective of a medical institution being able to trust a private site in terms of thinking about things like data leaks that have happened and continue to happen. Are there other alternatives or are there other ways that we could think to mitigate this?
I think it's a fair question. One of the things I always encourage people to think about when they're trying to decide whether or not to move operations into the cloud is it more secure than what you're currently doing? In many ways, what we're currently doing by managing our own servers locally is not necessarily any more secure than actually moving data out into the cloud, where you have teams that are constantly dedicated towards applying the most up-to-date patches and the best security practices. Does that mean that breaches won't happen? No, I still think that it comes down to best practice with respect to, encrypting data when it's at risk, making sure that the access and authentication protocols are correct. But there's also questions about, this question about trust that has to do with agreements more than anything else in terms of what the cloud service provider is allowed to do with the information that's been uploaded into their resource. Most of the time people are using AWS, for instance, it's local private, it's virtual private machines. So Amazon doesn't really have access to the information itself. It's really you just using their platform as a service. And so in that respect, there's a lot of control that you end up having. But as I said, no system is completely secure. It's really just a matter of where you think it's going to be best allocated in terms of your energy, in terms of management.
Great, thank you so much. This question really could be to either one of you. The questioner asked, what do you view the role of medical geneticists, and so physicians trained in residency for medical genetics, in the continued adoption of machine learning applications in genomics?
I'll take a shot at this maybe, and I'm interested to get Brad's perspective too. One of the things that people think about AI is that it's going to replace human expertise, and that couldn't be further from the truth. And that extends to, whether it's radiologists, pathologists, and medical geneticists. The human in the loop thing is so important that's why, the whole idea that we were just talking about with respect to the sick children and neonates, you got to have an expert overseeing this, because there are glitches. These algorithms, no matter how good they get, are always going to have imperfections, and human judgment, especially by people with expertise, is so critical. We're talking about, not just important, but potentially life or death decisions here. So we need medical geneticists. We need a lot more of them, actually. And what we're talking about now is just having this leaning on machines to deal with the data, and then the oversight, which is the fusion of this, which is the best possible scenario.
I think Eric hit it right on the head, to be honest with you. I mean, the notion that machine learning is going to be a substitute for human intuition, it's not going to be the case all the time. There are some places where I think you will see it with very clear application. I think image interpretation, for instance, like radiological services, where we're already seeing that systems can do as well, if not better, than humans. But you do run into these problems where what this does is that it frees up opportunity, it frees up time for humans to actually reason about the more difficult challenges. And so you're going to have computers that can do things 80, 90, maybe even 95% of the time, but you're still going to need humans that look at problems that are not actually being addressed by the machine learning or whatever type of artificial intelligence you've used. So you don't stop training people in specific areas. If anything, what you have to do is supplement their training to recognize that while the machines are going to provide some services to them, it's not going to provide all services. And it's in many respects, it's an evolution of a field. It's not the case that physicians are actually doing bench-based pathology work in order to facilitate their patients, right? This is something that they send things out to the lab, the path doctors or the pathologists are doing what they're supposed to do, and then the information comes back, and it's up to the clinician at that time to decide whether or not they trust what exactly it is that has been put in front of them and what to do next. No matter what, usually what happens, well, not no matter what, but usually what will happen with machine learning is that you're not going to just get a single best response. In a complex environment, you're going to get general rank ordering of what might be going on. And then there's going to be some further decision-making that needs to take place. And usually not all the information will be at the table. And so a computer will tend to say, you actually need to get more information. Here's what information I think you need to get. But the human now has to go do that. So the relationship, I totally agree with human in the loop, but it's going to be a symbiotic relationship that will continue to evolve over time.
I love that phrasing, the symbiotic relationship. That's perfect. Next question is for Dr. Topol. So you gave some beautiful examples of machine learning and AI in genomics in cancer and obviously in pediatric diseases or rare diseases. There was a question from the audience about where you see the initial implementation of machine learning for genomics in cardiovascular disease. And obviously this could be examples that you've already seen or areas that you think are ripe for this.
Yeah, I know it's an interesting question, Shannon, because the natural affinity of using genomics has been much more in cancer, rare diseases, not so much in other parts of whether it's neurodegenerative or cardiovascular. I think eventually we'll get there. The problem is that, that's where the multi-omics comes into play. These tissue-specific signatures, whether it's through RNA-seq and the epigenomics and all the others, that's what we need, in order to get a handle on the heart, the vascular system, and the brain, for example. Whereas what's so easy with cancer, now that we're kind of moving forward in the liquid biopsy space, is we've got a very easy access, whether it's for the tumor sample through a biopsy or through a tube of blood. And then with rare disease, we're basically looking at a whole genome sequence to understand that individual's clinical condition. So, there certainly is a wealth of knowledge about cardiovascular genomics. The problem is, though, we aren't nearly as cued in. I think over time, particularly in our challenge of this multi-omics, multi-dimensional layers of data. We'll get there.
Good answer. Thank you. The next question is for Dr. Mallon. Actually, there's a couple related to this, so you might both want to comment, but this one is directed to you first. They're asking about the differences between interpretability and explainability in machine learning, and which one, if there is a way to decide that, is more important for implementation in the clinical setting. And I will comment that there are several questions related to this thread.
Yeah, that's a hard question. Okay, let's take the semantics apart though. I mean, interpretability means that you actually have an understanding of what the machine learning model is doing. So the deeper a network, for instance, the more difficult it becomes to determine what the actual function is that's being computed to make the decision. That's different. So interpretability just means, can you actually peek inside the box and figure out what is going on? Explainability is a different situation. Explainability means that you have the ability to reason for the human. You can provide intuition into why the system is making the decision that it is doing. The most important, I mean, they're both equally important. Explainability, the reason why they're not necessarily mutually exclusive is that explainability means that the person who's making a decision at the end of the day has some intuition into why the system made the recommendation that it did. That's not the same as how exactly the function works, right? So you can, for instance, give me a model that says that it's going to give greater precedence to, imagine you're making a diagnostic workup and you are reliant upon both genomic information and phenomic information. And the system might come back and say, I'm making this particular decision or this recommendation because this region of the genome has the greatest influence on the model that was learned. That's not the same as saying why specifically or how specifically is that factor influencing the model. It's just known that if you change that variable, it's going to have some type of an influence. So now, so the explanation could be rely on this piece of information, but interpretability is, so what happens if you don't have the simplest of rule sets? Did the system create something that was an extremely complex representation that is not really representative of the way the world works? It ends up violating Occam's razor, for instance. Where you end up with imagine like a 10-layer deep neural network to explain some functionality when in reality a 1-layer or even a 2-layer would have sufficed. But the computer overtrained and kept jumping in layers, or you dumped in more layers to try to get like a little bit more accuracy out of the system. So, if you ever want to tweak what the model's actually doing, we're going to have interpretability. We're going to have the ability to allow what the computer has learned to merge with what a human knows should actually be going on, or have an idea of where the system is going wrong to allow it to be tweaked. But you're going to need explainability in order to get people comfortable with the use of the technology.
Fantastic. Anything you'd like to add, Dr. Topol?
Well, I think the simpler version would be, we'd like to have everything explainable. And what we're starting to see, not so much in genomics, but in other areas, is that using the AI to deconstruct the neural network. So to understand what are the features that it's seeing that the humans are missing or can't grasp. So we're just seeing the beginning of that. And what's interesting about AI is, for AI scientists, the answer for every AI problem is an AI solution. Now, the problem we have right now is that it isn't clear that we're going to be able to deconstruct and provide explainability for everything that AI can do. But that is what we would like to see. Wouldn't that be nice? And there's some hints that maybe we'll get there. So, I think it's interesting to follow this space. It's basically kind of reverse engineering the neural network to go backwards and find out what is it. There's some pretty good examples on the medical side, not so much yet in genomics.
Great, thank you. There's a question that's kind of near and dear to my heart, because at the center of it is around how do we nurture innovation? And something that was brought up, definitely, Dr. Topol, you even put a price tag on it in one of your slides, was how expensive some of these deep learning models can be to train. And so the audience member is asking, does this pose a risk to innovation in healthcare, especially if only large companies can afford to train them? And this individual actually is a CSO of a startup who's constantly worried about the cost of these models. So would appreciate your thoughts about how we can create a situation in which AI can be used by everyone for innovation and not just the biggest players in the world.
Yeah, this is really a central point. Brad touched on this, edge versus cloud computing. We wouldn't be in a position to use AI in genomics as one for graphic processing units. So these GPUs basically set the whole deep learning space into high gear. The problem is they're expensive. And if you go ahead and buy the hardware, and then, a few months later, there's a new version. And so then if you rely on the cloud, that's expensive. And, it's basically, my analogy is you're renting your house instead of buying the house, which is the right investment. And it's a mess. It's a mess because, the big chip manufacturers want to keep, coming up with better hardware. And the cloud is expensive, and it doesn't, all that money you put into that doesn't really get you any ownership. So this is a problem. We got guzzlers of GPUs, as I mentioned. Now, how do we get around this? Well, if we keep getting smarter in terms of coming up with, like the example I gave with the denoising autoencoder, we got to get less computing time. It's reminiscent of what Google did, where they used GPUs to reduce the energy consumption throughout their server farms. We need to do that in genomics. We have to get smarter on computing time and resources so we can essentially democratize this, because right now, this is a kind of a rich person's sport or science. It's very computational consumptive. And hopefully in the years ahead, we will see the GPUs come down in price. We'll see the cloud more competitive. Well, we need both, of course, but we also have to be using our ingenuity to make this cheap, which it isn't by any means today.
No, it's a fantastic point. Dr. Malin, did you want to add anything to that one?
Oh, I could add lots of things. No, I mean, Eric's right. There is just, you have decreased. One of the biggest problems we're running into is that we have had rapid decreasing in sequencing technology costs. And at the same time, we've had rapid uptake in the ability to use computational resources to analyze the data. So we've gone from something that was like $1 million to generate a genome down to the point where we're going to we're heading towards like $100. And but the costs are, the analysis costs are on the order of like 10,000 to 100,000, depending on the size of the study. So I totally agree that we need to have more efficiency in the computation, but I think what we're seeing, some of the stuff that my group does and other groups do, is create hybrid structures of computation, where you basically prototype on hardware that you've bought. And so that's like a fixed cost. And then once you're ready to do a large study, you then send it out to the cloud and you run it once, right? But you test. And it's really about understanding software engineering principles and how to build automated workflow pipelines. And this is something that some groups have become quite adept at, but it's something that we have to continue to train people in. I don't think it's quite as simple as just saying that, the technology is going to get better. It's really just about being smarter about the engineering to some degree.
It's a great point. One of the questions that came up, and I think this is really for both of you, is how do we meet the divide between the slow adoption by the FDA and they give examples of either polygenic or genomic-based approaches. And I think what's really under this question is also the idea of the algorithms, right, that are guiding these approaches as clinical tools beyond experimental or research purpose only, and the ability for these to become standard health insurance sanctioned interventions.
Ooh.
Go ahead, Brad, go ahead.
No, go ahead.
All right. Well, this is one of my pet peeves, Shannon, is that we have this wealth of knowledge that has been developed in genomics, and it sits in a separate orbit from helping patients and people prevent diseases and conditions that they otherwise might be at high risk. So we've tried really hard to implement that at Scripps, whereby we're using polygenic risk scores, particularly for heart disease, to help guide whether someone should take a statin, what is their overall risk for advising them. And we like to get that so that it's universally available. And as other polygenic risk scores are fully validated, that we get to implement those in practice for those people who want the information, not to force it, but for those who want it. Now, most people don't have that, and they're missing out on just years of data and just extraordinary amount of effort. So I really hope that we will. That's actually fundamental to that project that I outlined that Raquel's leading in our group, which is so we can make polygenic risk scores more widely available, very inexpensively across the board. There's so many now that are really been studied. I do think it's part of the dream of preventing illness that we talked about for decades, and we basically have never actualized. But I actually think that now that we know so much about common variants that are predictive cumulatively of risk, that we eventually will be able to help people who otherwise would be at risk to not have those conditions, or at the very least, sharpen our ability to manage or use medications that select which ones that might be of help. So I'm keen to move forward on this, but unfortunately, when I've talked at some of the genomic conferences, I get confronted with a lot of my colleagues and esteemed genomicists who think it's not ready. But I've seen too many patients feel really helped by this, so I hope we'll move in that direction.
The only thing I want to add to that is that there's an economics problem, and it's an unfortunate economics problem, which is that health insurers are slow to adopt new methodologies and new technologies for reimbursement. And so almost maybe 10, 15 years ago, Vanderbilt started doing prospective genotyping of patients who were at risk for cardiovascular disease and adverse events. And I don't take credit for this. I give Dan Roden and Josh Peterson and others a lot of credit for this. But we prospectively genotyped them so that in the event that they actually had some type of an acute event and then had to go on to warfarin or something like simvastatin, something that was a statin control. We had an idea of how best to tailor the medication to them from the outset. Now, the prospective genotyping was not expensive, but getting insurers to pay for this was almost impossible. And even after numerous demonstration projects were run, still trying to convince them to pay for that was way harder than trying to convince them to pay for whole genome sequencing of a cancer patient. When you know that the return on investment on this on a population-based level is the return is there. So it's not that it won't happen or it isn't happening. It's that it takes time to get these large industries to really evolve. But you need to continue to do the demonstration projects to prove that this is actually worthwhile to them, and that it would be a no-brainer for them to change the way that they do reimbursements.
Oh, really important point. There's a follow-up question. This is a very popular topic in the and they're asking both of you about examples of FDA-approved algorithms in the genomic space for diagnostics.
Ooh.
I'm not aware of any that are FDA approved.
I'm not aware of any either. Not yet.
There are many now that are cleared through 510K for images in medicine, but not that I'm aware of for genomic algorithms. Brad, I don't know of any.
I don't know off the top of my head, so I'm not going to speak out of turn on that.
Yeah, I do know of a really nice paper recently, and I'll post it in the chat, that actually kind of showed them, but it's exactly as you said, Dr. Topol, that many of them are don't have clearance yet. So it's kind of what's in the pipeline. And as both of you kind of mentioned, it's very dominant in what I would say is the earlier space, which is imaging, right? So radiology, that type of space. So I'll put that into the chat. Dr. Topol, there was a question for you specifically about the digital twin project.
Yeah.
Tempus, and they're asking to understand the difference between that and TCGA with respect to the data. And then this follow-up question is if it's possible to access the data from Tempus.
Yeah, well, we're working on trying to access that data. We haven't cracked into it yet, but we're working on it. You know, they've developed an incredible resource, but they haven't tapped into it. Theoretically, it will be of great value. So the point just to amplify or extend the comments there, we don't have a good example of an information infrastructure that would provide for medical digital twins. That is, where there's a wealth, ideally millions or billions of people, and matching up for every which way to predict the best treatment and outcomes. What we have relied upon until now are clinical trials. But as you well know, and even the best of clinical trials, maybe 5 out of 100 people benefit. So this is a whole new way to derive insight about what would be good for that particular patient. Now, because cancer has so much data, that's become nominated as the number one. And of course, the problem we have in cancer is the treatment and outcomes are often uncertain. They change all the time. So the whole idea, Kai-Fu Lee, who's a leading AI scientist in China, and I wrote a Nature Biotech paper, It Takes a Planet. And the reason we wrote that is we should be doing this for all of medicine, not just in cancer. That is, it's a way to help each of us in our species. Talk about a health learning system. I mean, this is the ultimate. So if we had, billions of people with all their data that was through federated AI and homomorphic encryption, which is tools that would keep the data at this place, whether it's the country or the health system, without any danger of privacy security breaches, that we could do this. And this is the future. I know it sounds, a little zany for some people, some of the listeners here, but this is a whole new opportunity. Like I mentioned, it isn't validated, but it makes a whole lot of sense. It's simple nearest neighbor analysis. And if you have, for each person, if you matched up several other people and you could look at their treatment and outcomes, whether it's for cancer or other conditions, it would give you another level of insight, but beyond what we have today, which is based on randomized clinical or prospective trials.
That's a great answer. Thank you. And thank you for your insight on that. Dr. Malin, there's a question for you, and this goes back to the cloud platform aspect and thinking about also the multi-party compute. So many of the cloud platforms are now imposing egress charges when data is moved out of the platform, so for the compute to happen elsewhere. Is that constraint taken into account when designing either distributed or leaner algorithms, and if so, how?
Yeah, it is. I mean, basically you have local compute, and then you pay for compute and you pay for bandwidth, right? So these are two factors that can be traded off in an optimization. That's not uncommon with federated learning models. And one thing I wanted to point out with the notion of federated learning is that it's not new. The notion of federated learning goes back almost 40, 50 years. So, I mean, the algorithms that have been designed, there are some new things to them, accounting for new types of statistical imputations and some distributed regressions. But a lot of the stuff that we're talking about, it really just requires engineering more than any real innovation in the mathematics and algorithms.
Yeah, fantastic, thank you. Question for both of you, and there's actually a couple of questions, so I'm going to try and synthesize it. This gets at the idea of, I would say, imputation, data augmentation, synthetic data, and there's concerns or just questions about what are going to be the issues in terms of either misrepresenting the general population or populations that are underrepresented, also about misdiagnosis or poor prediction. So if you both could kind of comment on, and these, as you both highlighted, these approaches are really needed because of how data hungry these algorithms are, right? And this also allows us to get around some of the privacy and other issues in terms of data sharing immediately, right? If we have synthetic data. So if we could have your thoughts just on how we can kind of balance that so that it's still meaningful and useful, and maybe what you see the limits of the synthetic data as being.
I'll jump on that real quick because it's kind of where we're still doing a lot of research. I think the issue with synthetic data is that you really have to understand the population you're trying to represent, and you have to understand some type of utility function. And I think Eric provided a great example of what you could do with augmentation when you know exactly what it is that you're trying to look for. Trying to create synthetic data as like an all-comers type of data set is a really challenging thing to do. I don't know if it's even completely possible. It's one of those grand challenges, in my opinion, that we need to have a better understanding of what the fundamental things are about people that or biological organisms that we're trying to represent in order to be able to generate synthetic data. But the thing is that if you understood everything about how biology worked, then you wouldn't need to generate synthetic data to pass it along. So there's a bit of a chicken or the egg problem here that we're dealing with synthetic data. I do think, though, that we have to think of synthetic data in two different ways. One is the privacy issue is one thing. And we should talk about like what types of things we're trying to actually protect against when sharing synthetic data, and what do we think it's actually going to be useful for? What are the warranties, and what are the guarantees that we can offer for it? But this other aspect, this notion of augmentation, is not about privacy. It's about filling in the cracks so that a machine learning model doesn't get lost and try to create a framework or a model that is not properly representative of the data. And I think that there's a lot of evidence over the last couple of years that this notion of augmentation is really one of the real wonders of what's going on in machine learning today, because it's facilitating breakthroughs in imaging informatics as well as genome science and lots of different areas. But it's still somewhat of a new venture. I mean, this is, if you wanted to create opportunities for graduate students to have dissertations for years to come, like this can go in many different directions.
Yeah, I would just add, Brad gave such a great perspective on this, both during his talk and just now. He's really been so thoughtful in advancing this. But we wouldn't need synthetic data if we had, the data sets from billions of people who are diverse. So this is basically a default that we've had to move to, because we don't have annotated data sets. We have largely European ancestry in genomics still. We don't have other ancestries adequately or symmetrically represented. So this is a great way to compensate for that. It's working, and it's important, but it doesn't substitute for the real deal. So, this is unfortunate. If we could go back 20 years, maybe we would have put more emphasis on this. And this is certainly one of the issues in the All of Us program of 1 million participants that's got more than half who are of underrepresented minorities. So ultimately, if they all do have genomics and multi-layered data, that will have another rich data set that will help be a cross-reference for synthetic data sets. So, it's great. I don't know what we do without synthetic data, but it's out of necessity.
Absolutely. There's kind of a follow-up question. So you focused on, both of you right now quite eloquently, the data component, but there was a question from the audience about how do we think about diversity, and I'll add fairness, when we're thinking about the models themselves and the algorithms.
Well, that's not so much a question. I think the how to think about this, there's the knowns, there are the things you're missing, and then there are the things you do not know that you're missing. And I think one of the problems that we run into is that you can develop risk calculators, or you can develop models that will look good in a population, but until you test it in another population, you will have absolutely no idea if it works. I mean, this is what happened with Framingham. But there's a calibration problem. So you actually have to ask the question of, do you go for diversity from the outset, or if you can, or do you try and build models and then test it on a diverse population to see if it holds? In many respects, you need both, right? But until you actually get the right samples to the table, right, you're kind of stuck in the situation where you develop the model you test to see if there's any holes, and then you recalibrate, and or you go and solicit additional individuals accordingly. But it's a tricky proposition, because you know that there's going to be holes. You just don't necessarily know where they are. And we don't necessarily know what all the factors are that are going to be influencing a model that we're dealing with, right? Because it's not just about race, it's not just about ethnicity, it's about, as I was alluding to, like socioeconomic status, it's about lifestyle. And so the more variables that you bring to the table, the more challenging it's going to be to have a single model that is representative of all of the population.
Yeah, no, it's a great point. I think there's, also the work now, that the number of tools that are out there on detecting bias and mitigating, but as we've seen with a number of high profile cases, it also depends on the metrics we're using and how we're analyzing it in terms of if we think that, an algorithm actually has been biased or not. So it's very complex, but great answer. Thank you. Thanks to both of you.
Well, but we should be clear that there is a difference between bias and fairness.
Absolutely.
Right? And bias just has to do with, is the data, trending in a certain direction such that it influences the model a certain way. Fairness is about, even if you recognize bias, can you correct for it so that you don't necessarily discriminate or provide opportunities that perpetuate a divide? And the challenge there that I think you run into is, to some degree, you may have to accept that we're not necessarily going to have the best system for a particular subgroup at the end of the day, if we want to try and bring the system into balance. Now, how to convince people to accept that situation, that I don't know, or if it has to be accepted.
Yeah, I would just add to Brad's point there. I think it is important. It's the humans that are responsible for the bias and the fairness, not the AI. Okay, so it's what we put in as inputs that's biased, and then the lack of fairness is how we apply that without, the thoughtful work to try to prevent lack of fair application. So, AI gets blamed unfairly, but it's really the I's and O's and not the algorithms and the models that are the problem.
3000%. The human context is critical. And I think that we miss that. It's very easy to blame the data or the algorithm and not recognize our roles in this. And this kind of goes back actually to a comment that both of you have made about the, education, training, the practitioners, right? Just as important. I know, Brad, you highlighted the NHGRI effort on that as well, that we have to make sure that we have representation across the board. There's a very popular question in the Q&A around the failure of IBM Watson Health. And people are asking if this is a prediction or a crystal ball about how long AI will be in the business of diagnostics and healthcare. So I leave that to both of you, however you'd like to comment.
Yeah, well, I mean, I've written about this a fair amount. I think what happened with IBM is they tried to say they could do things, and it was many years before that was possible. So when they established the partnership with MD Anderson for tens of millions of dollars at MD Anderson to say they could extract unstructured and structured text out of electronic health records, they couldn't do it. There wasn't any way. Only now are we starting to see that, like I mentioned, the project with Rades. So the issue here is that they had the right ideas, but they completely hyped it up and they sold it, and it was a bust. And there was a very important lesson for the AI industry, is that you got to have the goods. You can't be out there, they were blitzing, TV commercials about IBM Watson Health, and it was a lot of air. It wasn't real. And it's unfortunate because it could really hurt the field. Fortunately, I think what we're seeing now is a lot of things that they said they could do are actually getting done or starting to get done now. But all of us have to be wary about that because, these companies have different interests, and we have to know for sure before there's any major investment in something like this that it really works. So it's unfortunate that we went through that. And I think IBM regrets a lot of what they did, and hopefully we'll learn.
No, fantastic. I'm getting the nudge that we're almost out of time. This is an amazing discussion, but I do want to put in, there's a couple of threads in here around training and education. And there's one in particular from a resident physician who is thinking about how he can, quote unquote, get in the game in terms of this. So do you have any advice for, trainees, or actually I would say trainees of any level, about if they're interested in either learning about this or applying advice for them. So this is a question to both of you.
I think there's several opportunities. You know, I think it depends on what level of detail somebody wants to go into the field. You know, it's one thing to be knowledgeable. It's another thing to be a tool builder or an analyst. And I think you have to recognize where you want to be, first of all. That said, because the reason why I bring that up is that there are certainly training programs and tutorials that are more focused on the mathematics and the informatics itself. And so you can look for graduate training programs in that regard, or for postdoctoral opportunities. But then there's also things like the T32 programs that are focused on genomic medicine, where some of it's research, but some of it is really just in the application of genomics in the context of a clinical environment. And so those types of training opportunities are worthwhile for people who want to become practitioners more so than general hardcore genome scientists.
And I would just add, I can't imagine there's a more exciting area to work in. As opposed to the medical side, where it's a lot of regulatory stuff and it moves much slower. This moves so fast, and we're still at the earliest phase. And that's what people should realize. This is just starting to get going, and it has an immense future. So, you know, I think it's great to attract as much talent and enthusiasm as we can get into the field.
That's a brilliant way to close. I thank you both for your time and expertise. I know that this has been incredible. So many questions we didn't answer, but thank you both for your presentations today. And just as a reminder to the audience, we're going to take about a 20-minute break, and then we'll start up with the next session. Thank you.
Thank you.
Thanks for hosting, Shannon. That was great.
Thank you.
Hi, everybody. This is Trey Eidecker back with you again from UC San Diego. I'll be one of your two co-moderators for this official session 1 on algorithm development and machine learning approaches in genomics. And my co-chair.
Hi, how you doing? This is Anthony Philippakis from the Broad Institute. Dialing in here from my attic in Harvard Square. This is a very exciting session coming up. We have a great lineup of speakers, and Trey and I are very excited to be able to be here for it.
Great, thanks a lot, Anthony. And so without further ado, let's go ahead and introduce the first speaker. First speaker today is Dr. Jen Peng. He hails from the University of Illinois, Urbana-Champaign. He is an associate professor of computer science, and the title of his talk is Machine Learning Algorithms for Structural and Functional Genomics.
My name is Jen Peng. I'm currently an associate professor of computer science and medicine at the University of Illinois. Thanks so much for the invitation. And I would like to share of some of our research on structural biology and functional genomics, mainly from a machine learning perspective. So as we all know, a protein is composed of a sequence of amino acids. When the protein is presented in the solution in the living cell, it has to be folded into a particular structure. We call this native structure. And the problem of getting to know how after the protein is generated, how it gets folded into the right structure, the problem is called protein folding. And we have been studying this protein folding problem for decades. And why this protein folding problem is important? Why do we need to understand the structure? We all know that the structure provides a lot of insights on the particular location, particular sites, and some, for example, there's here I show one example. That is, this is a protein, one of my favorite proteins called protein kinase. What it does is to modify other proteins by adding a phosphate group, and this is a procedure is called a protein phosphorylation. And this procedure is original. We have no idea how this is done, and only after the structure is solved, we get to know. Okay, there's this structure has 2 lobes. Okay, 2 lobes, and in between there is a pocket where we host the molecule called ATP, where is one of the most important energy molecule, and also carry the phosphate group that is needed to be transferred into the substrate, which is a peptide or a fragment of another protein. And after this procedure, the phosphate is transferred to some of the residues on the substrate, and thus perform a molecular switch function and turn on and off another protein's function. Okay, so structure basically provides a lot of insights on the function. So therefore, the protein structure prediction is still one of the a very important task. It still remains one of the most challenging problems in computational biology. So the task of the protein structure prediction, and also sometimes people also call this a generalized version of protein folding, is that we're given an input sequence, amino acid sequence, and through whatever fancy computational approach you want to get, it's a 3-dimensional structure on the right side. Okay, and there are a lot of very successful prediction algorithm, including Rosetta, RaptorX, Atessa, and the newest version of AlphaFold. And I also show you here this progress of these different the prediction power collectively in the field across through the past few years. And here what I use is a metric that is used in the recent CASP competitions, where the entire community come together and evaluate their algorithms on some new proteins, unreleased proteins, and see whether predict structure has a certain level of accuracy. So I only showed you here the plot until 2018 because there's a huge improvement in the most recent CASP in 2020. But here, what's important is that I want to highlight that in the first, for example, in the middle of this maybe 20 years or so, we don't see much improvement. Okay, many of them improvement can be credited to the increase of the data. Okay, so we got a templates, we got a similar structures that we can use them to make prediction. But most recently, you can see there's huge improvement that is that greatly increase the performance of the structure prediction. So why is that? I just show you the two reasons I believe that to be true. One is that the machine learning algorithms start to become very powerful and can leverage a lot of data that we can make a reasonable prediction. Another is that we have a paradigm shift on how we do structure prediction. Previously, most reliable approach will be the template based, but now we're more into the regime there where we use co-evolution analysis and which rely on the contact prediction. Okay. So first, I would like to show the current status of protein structure prediction and what we have been doing in this field. And currently, there are two different approaches. As I just mentioned, there are two different approaches for protein structure prediction. So we're given first, but the first step is always the same. So we're given a protein sequence. We use sequence search algorithms to find all the homologous sequences and build this what we call as multiple sequence alignment. Okay, and traditionally the best approach, most reliable, is called template-based modeling, where we build some model, hidden mark models, or some kind of a sequence profile, and we search against this database. There's a structured database called a PDB, and we identify all these kind of possible templates, and we extract the distances, pairwise distances, or geometric constraint from these templates. You can think of the naive way would be that we copy the coordinates from the templates and put them together, blend them together. Somehow we build this backbone structure, and then we add a sidechain to the structure. Okay, but this methods has been very well, although it's very reliable, but it's hit to the some bottleneck that we can, there's an upper bound, we cannot further improve the performance of template-based modeling for quite a few years. And most recently, these years, the new paradigm will be this what we call a contact system modeling, where we rely on this information within this alignment. So we extract something like a correlation between or among these different sites, and we predict whether these two residues, two amino acids, will be spatially close, okay, according to the correlation in the alignment. So this is called a co-evolution analysis. And also with the deep learning models, we'll be able to predict a lot of properties, a lot of constraints regarding the pairwise residue interaction. For example, whether they're close by or their orientations to be meaningful. And with this constraint to predict it, we can use them to optimize the backbone and obtain some really good initial structure. And further, we can also add a sidechains. We perform some type of structure refinement. Okay, so here the core concept is based on this co-evolution. So here I show you why this is a good intuition. So basically here we have after we build this multiple sequence alignment, we look at these two columns. And if we have a way to evaluate the strength of the co-evolution of these two sites, or for example, here the EIJ, this is a term that is used to quantify the choices of these, for example, two different amino acids, and this absolute value will give us the strength of whether they like to co-occur or they don't like to occur together. Okay, and this is biological meaningful. For example, consider on the right the structure. We have these three residues, and these two, the red one and the blue one, tend to be interacting with each other, and because they're spatially close by. And when we mutate substitute one residue to another amino acid, the other one you would like to also be mutated or substituted accordingly. And on the other hand, the remote one, for example, the orange one, will be far from them. So the choice of the orange one usually has nothing to do with the choice of those two. So this is a basic concept. So essentially, we have this assumption that evolution and structure are this constraint should be similar to each other in terms of these residue interactions. Okay, so here's a model usually people do, like this is how we actually capture this kind of interaction. So the naive model people use is based on the, for example, this PWM or specific scoring matrix. These are based on assumption that each of these location is independent. Okay, and so here we have a different way to write this, and This is through an energy-based representation. So we have the single potentials, which means that the choices of these letters on the ice position has nothing to do with the other sides. Okay, but this pairwise-based approach actually gives us a way to learn the coupling, this co-evolution strengths. Here we introduce another term that is a sum over all these pairwise potentials, which gives us co-evolution strengths. And this particular formulation is very well studied in machine learning, in statistical physics, and in high difference statistics, where it has different names: Markov random fields, Ising model, or undirected graphical model. Okay, so to solve this this model, which is that we want to learn these strengths from the data. For example, singleton potentials, pairwise potential, where this is mostly critical one, we need to solve some problem that are related to the maximizing the data likelihood, which is shown here. And here there's a partition function, which makes the problem computationally very hard. So there are a lot of approaches have been developed for approximation. So there are mean field approximation, Gaussian approximation, pseudo likelihood approximation. They all give different versions of the evolution, co-evolution analysis. And people have shown that these are much better than the normal correlation analysis, which is for discrete variables. We call them the mutual information here. Okay, so what is mostly exciting about this is that now we have the way to show that the correlation between the size, but it's also very noisy. So what we have, my group has really changed the field, I think, changed the field and initiated this introduction of deep learning to the field, is that we leverage some of the intuition we got from natural image recognition. For example, here, this is a classic deep convolution neural network. It is very helpful to recognize imaging patterns. For example, these different patches. We can organize these different patches in a hierarchical way to give some output. Okay, and here we also treat this protein, like for example, co-evolution data. Co-evolution, we also call evolutional couplings, as image. Okay, we also look at the local patterns in this evolution coupling matrix, and we apply this hierarchical deep learning model to put all these signal together in a hierarchical manner, and then predict the distance contact map. Okay, this can be binary, which means that whether they're close by, these residues are close by in space, or we can just predict the distance. Okay, and this is our model. So we build this model, we learn from the co-evolution. You can see this is a very noisy signal. Okay, and then we go through all this hierarchy. This is a hierarchical neural network, a deep convolution neural network, integrate all kinds of features. And on bottom, you can see that we basically, this model denoise the original, the co-evolution patterns, and also generates the contact map looks very similar to the actual contact structure. Okay, so here is another example. This is actually the distance matrix we see. Okay, from the native structure. This is the program we use to generate the input for the neural network, and it looks very. You can see there's some pattern, but very noisy. And this is the version after we apply the deep context, the neural network to the sysampred. So what we got here is that we use the contact map to help fold the structure. So we tested it in CASP12. You can see that it generated much better structure than the original input, and our program is also ranked among the top in CASP12 a few years ago. Okay, so why does machine learning, deep learning work for this particular problem? So we actually perform some visualization. You can see that here we take the first layer filters and visualize into 2D, and we color them according to secondary structure. You can see that this parallel or anti-parallel patterns of beta strands are all these kind of diagonal or anti-diagonal patterns. And for helical directions, we see this kind of spaced patterns, which corresponds to the gap, corresponds to the space between them, corresponds to the number of residues in each turn of the alpha helical. So this corresponds, this visualization shows that with the deep neural network, we actually learn something that is meaningful. And at a higher layer, these patterns are organized into higher, like more complex structure motifs for structural prediction. So, okay, after this many groups have developed different approaches for example, Raptor X, AlphaFold, and Rosetta. They all based on the similar intuition, building the extracting the patterns from the coevolution data and predicting the residue interaction and some other angle properties like inter-residue angles, and they have been also shown to be successful. But what's mostly interesting is that what happened last year, that in Casper-14, DeepMind's approach, they have some component related to the previous, what I just described, but they greatly improve the performance. Okay, you can see that the GDT, which is score between 0 and 100, 100 means perfect prediction, and the AlphaFold is able to give a score close to 90, and this 90 is about the variance between this, about the consistency between the two structures solved by the two different groups. Okay, so here I also show you down there. There are very challenging two structures. I tried all different approaches and existing algorithms. These are the structures from SARS-CoV-2 virus, and all existing structure prediction algorithm cannot make any reasonable prediction for these two. Basically, we just got a random prediction, but AlphaFold 2 is able to generate a prediction that looks almost perfect. Okay, so which I think is very impressive. Okay, so in the rest of this talk, I'll move a little bit away from structure prediction, but focus on the function prediction. So beyond the structure prediction, what we really wanted to know, like you understand, is actually the protein function. That is, we're given a protein structure or sequence, we want to understand its function. It has a lot of applications. For example, we can use it to improve, if we have such a model, we can use it to improve binding affinity of antibodies, optimize fluorescent proteins, or even improve the specificity of gene editing. Okay, so what do we need to have such a model from a machine learning perspective? So essentially we want a model that takes a sequence as input and give an output, which is a function value. Okay, so the model that is useful for function prediction has there should be some requirements. For example, we need the model to be sensitive enough to differentiate the function level of very similar sequences. Okay, because sometimes we know introducing an important residue will completely change the function. And we also need to model the non-additive effect, epistasis, and changes of these different residues may have interactions. And we also need this model to be good enough to generalize to unseen sequences and mutations. Okay, so my group has developed quite a few different machine learning based approaches for modeling for modeling the protein function from sequence. For example, we have this recurrent neural network versions. We also have the model that takes both the protein sequence and the substrates as input using the convolution neural networks. And we also have applied this different approach to different applications, including phosphorylation, protein RNA binding, and protein drug binding. Okay, so all of these different applications requires a lot of data to train the model. Okay, so but in practice we don't have a lot of data. The label data is expensive to get. So there has been a lot of ideas in the machine learning community we can borrow that can be applied here. So one idea is that we can use unsupervised learning to learn good representations using language models. Okay, so these data are very easy to obtain. We don't have labels, but they present some kind of good representation of the protein or the sequences. Okay, so basically the idea is that we have the sum of the sequence of the residues we have seen, amino acids we have seen, we want to predict what we will see next. And there are other models like transformer models, which is more complex, but is also able to learn to make such predictions. So these approaches can be trained. These models, large models, can be trained on the PFM or UniProt with a lot of sequence, but without any labels related to their function. Okay, but the drawback of these approaches are too general. Maybe they will just capture secondary structure, solve an accessibility, some of this basic grammar of this protein sequence, but will not specific enough to reflect what the special function we want to study. Okay, another idea we take is similar to what I have before, is that we can learn from evolution. So here we can build the sequence alignment And we assume that all these sequences have some native function, and they have or passed the selection step during evolution. So we can treat those as weakly labeled data. We know they have survived through the evolution. We know they can be treated as a positive data somehow. And these data are not a lot, right? We know that there are not a lot of data to train very complex deep learning model, but they're good enough to extract good features. So what we have done is that first thing we did is that we can check whether this kind of evolution patterns actually correlates with function. So on the left, we have some experiment data from deep mutational scanning, like pairwise deep mutational scanning. And on the right, we have this model we have shown you before to compute the co-evolution pattern. And we actually check whether they correlated. And in our different sites, they have different correlations, but on average, if we have a lot of homologous sequence in the alignment, we tend to have a stronger correlation. Okay, so this gives us some insight that we can actually use co-evolution patterns to form features. Okay, so this is our model. We call this ECNet by integrating evolutionary context, global context, and local context for protein function prediction. So on the top, we use a global model, language model, to extract the sequence representations. And also on the local, we have this model that it learned from multiple sequences. Those are homologous sequences, very similar sequences, close sequences, and we use them to learn these specific and these features that are sensitive to mutations. And then we use our favorite model to integrate all these features together, and so that to predict the protein function. So we have evaluated this on a large collection of deep mutation scanning data. These are single mutation data sets, and we perform rigorous cross-validation and show that it works much better than the previous unsupervised versions, such as the EVFold or deep sequence, and also better than the supervised model that is developed by the machine learning community. Okay, so we have also tested the generalization of this model to high order mutations. And you can see that on the left, we tested on the GFP, and seems like with single and double mutation, we'll be able to predict a very high order mutation, which means that maybe the pairwise, the high order interactions may not be that important. The pairwise might be good enough. And on the right, we also tested that collect the literature data on these resistant TEM. This is beta-lactamase, and show that our model is able to differentiate these antibiotic resistant alleles against the random alleles. Okay, so finally we apply this to actually engineer the inhibitor resistant beta-lactamase. So we take the structure, the sequence structure. We use our ECNet to design a collection of mutants with high order mutations, and we check whether they are resistant to a very important antibiotic compound, ampicillin, and we tested across different concentrations. And we have here, I also show here we have two different models, and we also have the positive controls. There are very, very handful of positive controls in the literature. We collect only, we examine all the literature, only collect a very few number of them, which means that this is a very hard to engineer protein. And we did we replicate this experiment multiple times and show that actually our hit rate is really pretty good when the concentration is high. Okay, so finally, I would like to thank the audience, thank you all, and also thank my students and my collaborators and all the funding agency. Thank you very much.
Okay,
Thank
great.
you so
Thank
much.
you very much, Zhentang.
Yeah.
So the way this is going to work in the sessions is we'll have a very brief Q&A after each talk, and then we'll have a lot of time at the end of the session. We'll have 30 minutes at the end to have a broader discussion. So just quickly, let me ask one question from the audience. How did you decide the size of the images in the coevolution, presumably as input to the convolutional neural networks you talked about? Would different image sizes cause learning of different relationships between correlated patterns?
Right. To prepare the input, we first compute the evolutionary couplings and summarize those values for each pair of for each pair of positions. So essentially, if you have a protein with length L, you got L by L matrix input. Now, of course, there's a lot of engineering needs to be done. For example, because we don't have large like a RAM GPUs, we have to cut the proteins into meaningful domains. So there's in the current version, we mainly consider the intra-domain interactions, not intra-domain interactions. And that will make the engineer much easier to do. And I know there are a lot of other groups also consider intra-domain interactions and protein-protein interactions. And there are some other like kind of input needs to be prepared, and also the evolution and patterns needs to be formatted in a different way. So it's essentially this is in practice we mainly consider the whether we can actually implement the algorithm is like run them with sufficient with a given resources, not related to whether we want to. It's not that we want to cut them into pieces. It's mainly an engineering consideration.
Great. Another question is, what is the success metric used for 3D structure prediction? Maybe you can say a little bit more about how it works.
Yeah, so usually there are quite a few different metrics people use in the field, but what they're they do are pretty similar. So given predicted 3-dimensional structure and a native structure, what usually the first step we will do is superimpose those two geometrical objects. There are a lot of algorithms to do that. After doing that, we just check whether the deviation between the predicted residue position. Usually we consider, for example, the backbone atoms, C alpha atoms, and how far that each predicted C alpha atom is from the native atom, and then we compute, for example, like we can set a certain threshold. Like maybe we can consider like for example 1 angstrom to be meaningful, 2 angstrom to be meaningful. Then we can summarize the fraction of the correctly predicted to the percentage of the corrected predicted residues. And so this is usually people do. For example, GTT score I mentioned here is computing this way by averaging multiple threshold. There are also some other metrics people use. For example, comparing their pairwise distance, but all of them have the similar manner. So usually people summarize that to a score between 0 and 100, or 0 and 1. 1 means perfect, 0 means not meaningful.
Okay, great. So to stay on time, let's move on now to the second speaker. And again, we'll have lots of time after the session to discuss more.
Excellent. So our second speaker will be Sarah Matheson, who's an assistant professor of computer science in Haverford College.
Hello, everyone. My name is Sarah Matheson. I'm an assistant professor at Haverford College in the Department of Computer Science, and I'm very happy to be here for the Machine Learning in Genomics workshop today. And specifically, I'm going to talk about generative adversarial networks and their use in population genetics. But more broadly, I want to give a sense of where we're going in machine learning and population genetics, and also kind of the shifts that have happened over time in the field. So one of the big questions in population genetics is, how do we go from all of the simulated data, all of the sequence data, and actually learn something about evolution? So here I have this big matrix of zeros and ones representing a bunch of different samples and a bunch of different SNPs for these samples. And say I want to learn something like recombination hotspots or something about the recombination landscape. But I don't want to do this just for these particular, this particular sample, or just for this particular problem of recombination. I want to do this more generally in any species that I'm interested in, and maybe any evolutionary phenomenon that I'm interested in as well, say heritable traits or diseases, admixture, natural selection. I don't want to have to redo the entire method every time that I change the application or change the species. And so if I were to think about what are the properties that I really want for this type of method, I would definitely want it to be fast and flexible, and maybe I would want it to be a machine learning method. So I think there's been several shifts in population genetics recently, and one of them was toward machine learning in around kind of 2010 timeframe, and then a second shift that started to move away from summary statistics. And then I'll talk a little bit about one of my ongoing projects in my lab about using generative adversarial networks and adversarial training more generally to create simulated data. So what about this first shift toward machine learning? So I think one of the papers that really highlights this nicely was in 2013. This paper that was trying to identify regions as either neutral or under natural selection, so a binary classification problem. And one of the observations, which has been known for a long time, is that selection distorts properties of genomic data. And in particular, this is highlighted in the site frequency spectrum, which is a common summary statistic. And so in these figures, the blue line here, it shows a neutral region and what the site frequency spectrum would look like. And these red curves show what it would look like under natural selection in a variety of different scenarios. So could machine learning, which is known to be good at pattern detection and highlighting and picking out subtle signals, could it be used to actually detect natural selection? And so the authors showed that it could. And their method that they used was a more classical machine learning method of the support vector machine. And broadly, how this works is that neutral regions are simulated, in this case of natural selection, shown in red here, and then regions under selection, shown in blue here, are simulated as well. And the idea of this model is to try to identify some boundary or some barrier between these two different types of regions. And if we're able to fit that boundary very well, then if a new data set comes along, say a real region that we're trying to see if it's under selection or not, then we can plot it in the statistics space and actually see which side of the boundary it's on. So in this case, we would probably identify this region of real data as predicted as under selection. I think this is a really nice example of how machine learning had started to be used, because it was very good at finding signals from summary statistics. But the issue arises in the case when we don't really know what summary statistics are good, or maybe we have a lot of summary statistics available. Here are a bunch of common ones I used for a project around 2016. And we don't really know if we change our problem of interest, do we need to change our summary statistics, or do we need to invent new summary statistics? So one usage of machine learning is really to identify important features or distill down combinations of features that will be really useful for a problem of interest. So what we decided to do was feed all of these summary statistics into a deep learning method. The deep here just means, these multiple layers, and actually infer both selection and population size changes. So here represented as this simple bottleneck model, but jointly inferred along with natural selection. And the idea was that this deep learning method could actually distill down information within these summary statistics, creating groups of summary statistics or features built out of summary statistics that would be informative for these parameters. So I think there was a lot of activity in machine learning around this time, but one of the drawbacks was that it still relied on these summary statistics. And so then there was a second shift away from summary statistics toward trying to use this raw data. And part of my motivation for moving away from summary statistics was actually to not have to change them for every application and not have to think about inventing new ones for every single new problem that I'm interested in. So for this, I think several groups drew inspiration from convolutional neural networks, or CNNs. And these have been developed in a lot of the image recognition and classification literature. And very, very broadly, one of the key ideas of CNNs is that these little filters could pick up on different aspects of these images. So say, for example, maybe this filter would learn to identify beaks or feathers. And then if those filters sort of lit up in classification, then we might be more likely to conclude that this image is of a bird. So the idea was that these learned filters could be used in genetics to try to find out which aspects of the data were really informative for certain parameters of interest. However, there are a few issues in this. One being that these filters are really specialized for images, but they are not really specialized for genetic data. And the second issue is that for, say, an unstructured population where the order of the individuals is not really informative at all, we don't want to encode that into our network architecture. So for an image, if we imagine just sort of shuffling all of the different pixel rows of the image, it wouldn't mean anything anymore. But for genetic data, we want to say that's actually okay, that's actually the same data set. So I'll focus, there's a few methods during this time that worked on CNNs for genetics, and I'll focus on this second one, where we were trying to create an architecture that would actually sort of respect some of the properties of the underlying data. So for this, we looked at our raw data as, again, a series of zeros and ones in a matrix, but the samples or the rows are now exchangeable. And we fed this not through these square filters, but actually used filters of height 1. So these filters actually only spanned one haplotype or one sample, and thus they didn't encode any information about the ordering of the samples in this network. And then we did that for several layers to make sure that we weren't assuming anything about the order. And then toward the end of the network, we actually collapsed along all of the rows. And so that would give us something that was also a permutation invariant. So there are lots of different permutation invariant functions we could choose here. Sum, max, average are all permutation invariant. And this allowed us to kind of distill down some of the information that had been learned in the previous layers. And then finally, we can infer some evolutionary parameter that we are interested in. So this network was able to work quite well on a few different types of problems. One example is actually looking at recombination hotspots. So a binary classification of either a region inside a hotspot or not. And we plotted over the training iterations, we plotted the accuracy. And we found that for this blue line here, this was the exchangeable architecture or the permutation invariant architecture. We were able to get higher accuracy than if we just used some of the more image-oriented CNNs shown in these other curves here. So really explicitly encoding the nature of the data was able to help us perform better in this network. So this was, there's several different CNN methods developed during this time, and I think it was a huge step forward for the field. But one thing I've kind of glossed over is that we still need simulated data for these machine learning methods. And actually, it's very important for machine learning methods in genetics to have simulated data as the training data, since we don't really have any real examples where the evolutionary history would be known. Or in other words, we need to use supervised learning because we don't really, we need to use supervised learning, and therefore we need the labels, and we don't really have the labels of any real data. So we really rely heavily on that simulated data. So I became very interested in developing better simulated data. And this was inspired by several times when I tried to create realistic simulated data and gave my sort of best guess at the parameters, and I still was not really able to accurately capture some of the features of the real data. So here are a few examples where the real and the simulated data sets look very different, and here as well, even when I tried to put in sort of reasonable parameters for these simulation programs. And as a broader point, the role of simulated data in population genetics can really not be overstated. it is useful not only for training these machine learning methods, but for validating methods, developing intuition. It's extremely important. And to that effect, many different simulation programs have been developed over the years. And two of the most popular ones right now are SLIM and MS Prime. And these programs work very well at recapitulating evolution, but they do require many input parameters. And so it can be very difficult to really identify what are the best ones. And here are some examples where this didn't really work. So I was interested in trying out generative approaches, and specifically generative adversarial networks. And the idea behind these GAN algorithms is that our initial guess at whatever type of simulation we're trying to come up with is probably going to be pretty poor quality, right? If we just guess, we're not going to be able to develop a really good simulation right off the bat. So to draw an analogy from the art world, say we are a forger and we're trying to make sort of fake artworks and, in the style of famous artists. And so if we just try this and we're starting out, we're not very experienced, and we just try this, our fake data might look like this. And so now it's pretty easy for, say, an art critic to determine, well, one of these is fake and one of these is real. So say we go off and we say, okay, well, that wasn't very good, but we try to train more and we learn more about art, and we try again. And maybe this time we're a little bit better. So we try this example on the left, but it still doesn't really look like a real Picasso on the right. So we go off and we train more and we learn more about art, and we try again. And I'm not sure what these look like to you, but to me, they look pretty realistic. But it kind of turns out that both of these are suspected to be fake Jackson Pollocks. Okay, so kind of being a bit more detailed, if we were to think about what a GAN architecture really looks like, it has these two components. It has this generator, or we can think of that as a forger, that's trying to create these synthetic examples. And then we have the discriminator, or we can think of the art critic, that's actually trying to identify this real versus fake. And an important part about the discriminator is that they have to make a binary classification at the end of the day, whether that example is real or fake. And as part of this feedback loop, based on that information, the generator tries to perform better. And so eventually, both of these entities will become better over time, with the generator creating more and more realistic data. And then because of that, the discriminator has to get better too and identify these subtle differences to try and identify which are real and which are fake. So this is kind of the broad idea, and this has been used in images before. But for population genetics, there's quite a lot of modifications we need to make to really get this to work in a new system. So what we did is designed a new GAN framework for population genetics, which we call PGGAN. And the idea is that we still are feeding in generated data and real data into the discriminator. And training the discriminator that way. But instead of the generator going right to simulating all these zeros and ones, I actually wanted to include an evolutionary model as part of the generation process. So instead, the generator needs to choose these parameters of, say, an evolutionary model, say these population sizes, N1, N2, N3 in this example, and then feed those in to some evolutionary simulator and then create the generated data. And so because of this unique generator architecture, we can no longer really use backpropagation or a lot of gradient descent style approaches because we don't have the gradient anymore. So to overcome this, we actually used a simulated annealing algorithm to select the parameters that we would feed into the evolutionary model. And the idea here is that we start out with being able to rapidly move across the parameter space in terms of our evolutionary parameters at the beginning. And then as this temperature cools down, we're able to make more small refinements to our parameters until at the end, we should hopefully have parameters that generate data that is as realistic as possible and confuses the discriminator. So this was our generator, and then for the discriminator, we chose to use a CNN. And for this CNN, we actually expanded on the permutation and variant CNN I talked about earlier to include multiple populations. And the idea here is that we want to include a richer set of evolutionary models. So we need to handle multiple populations. And within each population, the samples are permutation and variant, but between populations, they are not. And then after several layers, convolutional layers and filters here, then we collapse along the rows and then concatenate the two populations together. And then instead of the output being a evolutionary parameter of interest, it's actually this binary output or probability of being real or fake. So we have this sort of binary classification problem. So before I talk about the results, I wanted to say just a little bit about what can go wrong in GAN training. GANs are notoriously difficult to train, and in part because there's two optimization problems, the generator and the discriminator, and they're adversarial, so they're competing against each other. And so one of the things that can go wrong is that the discriminator classifies all data sets as real. So across these training iterations on the x-axis here, if we look at the accuracy on real and fake data, the discriminator is 100% accurate on the real data, classifying everything as real, but 0% accurate on the fake data. And this is also represented in the loss, where the generator loss is not very low. It's not getting any positive feedback. It's just everything it does is classified as real, so there's no incentive for the generator to improve. So this is definitely a major thing that can go wrong. In contrast, looking at examples of successful training, we see that in the beginning, usually the discriminator is very accurate, and then it reduces its accuracy over time to around 50%. And then at the end, it's really often confused and not, even though there's some back and forth, it's typically not able to really accurately determine real or simulated. And at the end, the losses are balanced, meaning that the generator and the discriminator are both really working hard to do their jobs. So one way of evaluating this type of method is to look if there are some features of the real data that are accurately being captured by the simulations. So we looked at summary statistics here as a sanity check that our method was working. And so for one example, we looked at just a single population, and here's CHB, which is an East Asian population. And if we just use a constant population size, then we weren't really capturing the real data very accurately, or these green and gray curves were not very close together. But if we used a more sophisticated model that actually included exponential growth and some size changes at different times, we're actually able to much better fit the data. We also looked at a two-population model of modeling the out of Africa event with YRI and CEU, an African and European population. And we're also able to really closely mirror the summary statistics here. And then we are also able to fit this model and just look at the parameters as another sort of sanity check that we were getting reasonable results. And we do see the out of Africa model, out of Africa bottleneck and re-expansion here in CEU, as well as some migration post-split. So this was encouraging that this was broadly in line with the existing literature. So a little bit of where we're going with machine learning. I think there's many different opportunities for PGGAN in particular, especially for studying understudied populations where we don't have a very good idea of what the default parameters would be. And then also overcoming this fundamental imbalance since we have unlimited simulated data, but limited real data. And I think more broadly, we really need to keep the data in mind and not just take off-the-shelf methods from other fields, but really think about what methods will be most applicable for our data and what modifications and new algorithms do we actually need for genetic data, such as this permutation and variant architectures or simulation generators that don't rely on the gradient. I think we really need machine learning methods to be more interpretable. We don't really know what these CNNs are learning yet. And I think there's a lot of opportunities to combine machine learning and evolutionary modeling. So it's not one or the other. And finally, I would say that I think we need to think more about moving away from simulations too, and thinking more about unsupervised learning and learning from the real data directly. And with that, I wanted to thank all my collaborators and funding, and thank you for your attention.
Thank you so much, Dr. Matheson. Wonderful talk. So I'll start with a few questions by our audience. So first, you said that you needed simulated data for training supervised models. Are you also exploring unsupervised models?
Yes, and I think that's a great question. And I think that part of where the motivation for the GAN framework came from is actually this exact idea, is that we're not just using purely simulated data that we kind of make up based on our best guess, but we actually try to use the GAN to generate better simulated data. And thus the real data is actually fed into the GAN, and then the simulated data tries to match that. So I think that that's kind of a hybrid approach in terms of it's not purely unsupervised, but we're trying to use the real data directly instead of at the very end after we've already trained on purely simulated data. I also think there's a few other groups in this area that are working on using unsupervised learning more directly in terms of visualization and clustering and things like that, that would actually be really purely unsupervised without any training data at all. So I think there's much room for improvement, though. I think I would definitely encourage people to kind of think about this area of unsupervised learning in population genetics.
Great, thanks, Sarah. I'll ask this last question by Zhang Yun. For the SNPs data as input to the convolutional neural network model, they assume 0 and 1 refer to the number of minor alleles. Have you tried the 0, 1, 2 type input in your models, and how to optimize the stride in the CNN model?
That's a great question. So yeah, 0 refers to the minor allele and 1 refers to the, or 0 refers to the major allele and 1 refers to the minor allele. You can also, if you know your ancestral derived state, you can have 0 as the ancestral state and 1 as the derived state. That would also work. I think if it would be interesting to try other approaches. You can also try like a -1, 1, and then 0 is for missing data, which I think could be better in cases when you the quality is less good. In terms of increasing the number, I mean, usually we really see biallelic SNPs, and that's why you don't sort of see higher numbers, but I think there's no reason not to include them if that's better for your model. I think you just have to be really careful, because we're using ReLU and all these sort of different activation functions, you need to be careful about where is your 0 mark and what are you really assuming that, you don't want everything to be positive if those are the things that are going to be sort of filtered down your network. So you kind of have to be aware of that.
What do you think, Anthony? Shall we do one more, or shall we move on?
Well, there's a really good question, so maybe we'll do one more. So what type of loss functions do you use for imbalanced data, and do you apply any upsampling for unbalanced data?
That's a really interesting question about this sort of data imbalance. For us, we try to simulate the exact number of real data sets we have. So we kind of get around that a little bit with the balancing of the data. I think generally we use this sort of like binary cross entropy loss functions for the GANs. I can talk more about that in the main Q&A. But I think that in general, there is this fundamental data imbalance, right? We have unlimited simulated data, we have limited real data. And so I think that needs to be further explored, but it's not something I've done yet.
Okay, great. Thank you, Sarah. So let's move to the final speaker of our session 1. It's my pleasure to introduce Dr. Christina Leslie from Memorial Sloan Kettering Cancer Center. Her talk is titled The 3D Genome and Predictive Gene Regulatory Models.
Hi, my name is Christina Leslie. I'm at Memorial Sloan Kettering Cancer Center, and I want to talk to you about using machine learning models together with 3D genomic data to train predictive models of gene regulation. All right, so as I'm sure you're aware, gene regulation involves the collaboration of transcription factors that bind at the promoter as well as at the enhancers that can be quite distal from the promoter, and DNA looping that brings enhancer elements in contact with the promoter and leads to upregulation of expression of the transcript. And for quite some time, we've used 1D epigenomic data, information on where transcription factors are binding, information about chromatin accessibility and histone marks to map the presence of candidate enhancer elements. But we haven't used, until recently, the 3D connectivity of these elements. Okay, so why do we want to try to predict gene expression? Well, gene expression is important for the function of cells and for understanding what cells do in different states. And this is information or data from one of my own papers, but there are many such studies where we're looking at chromatin states of, in this case, CD8 T cells and functional T cells and T cells that progress to dysfunction in tumors. Okay, and what we can see by looking at this chromatin accessibility at specific loci, so this is the locus that encodes PD-1 in the mouse. PD-1 is an immunotherapy target, and this is the locus of an important effector cytokine interferon gamma. And what happens as T cells progress to a dysfunctional or exhausted state, there are chromatin accessibility changes at these loci, different from when you get effector or memory functional cells, okay? And these changes somehow encode the gain of expression of PD-1 and the loss of gene expression of interferon gamma. And we'd like to understand this and model this. For NHGRI, it's also important to link genetic variation to gene expression. Okay, and this is an example that's not from my lab, it's from a recent paper trying to understand the association between genetic variation and gene expression of a particular gene, okay, BLK here, as well as doing association of genetic variation with chromatin accessibility and teasing apart how changes in the DNA sequence can change accessibility and chromatin state, and that can change gene regulation. Okay, so ideally, we would like to have machine learning models that could automate this process. In my own lab, we've done a lot of work in this domain, trying to use models to predict gene expression or fold change between different cell states from the sequence content and the accessibility or activity of regulatory elements. And we're doing this not to predict per se, but in order to decipher gene regulation, figure out what the regulators are, figure out how individual genes are regulated. And the missing information in our models so far has been the connectivity information, the connectivity of promoters and enhancers. So what I want to show you today is using 3D interaction data in order to model gene regulation using graph neural networks. Okay, so the data we're going to use is chromosome conformation capture data, such as high C. Here's a picture of high C. The basic idea is that you're cross-linking proteins to DNA in situ. You do a restriction enzyme digest so that you're cutting up the chromatin, you ligate and pull down, and you get paired end reads where the read pair maps a contact. So the pair of reads can be distal in the 1D genome, but they had to be close in a cell in the input population. And this data allows us to build a contact matrix that gives us information about 3D proximity of genomic regions. All right, and we can look at these contact matrices at different scales, at a genome-wide scale or chromosome-wide, but we're more interested in these domain level and loop level and individual promoter enhancer interaction level information. Okay, so if you zoom in to these maps close enough, you can see organization-like topologically associated domains, and within these TADs, you can see some loops between genomic loci. Okay, methods matter. So in our lab, we've developed our own statistical approaches to infer interactions, significant interactions directly from count data. And we call this method high CDC for high C direct caller. So essentially what we're trying to do is estimate a background model from the count data using negative binomial regression. The covariates that are important are the genomic distance, as well as features of the interaction bins, such as mappability, number of restriction enzyme sites, GC content. And when we fit our model, we can observe the counts and decide how surprised we are to see a high count and assign a p-value or a z-score. Okay, and this latest version, high CDC plus, is in revision, hopefully coming out soon. And when we can do that, we can start to see interactions that are of interest to us for modeling gene regulation. So this is data from Danwei Huangfu's lab in collaboration for our 4D nucleome project that also involves Effie Apostolou. And what we're looking at is a progression of guided differentiation of human embryonic stem cells towards insulin secreting beta cells. Okay, and looking at these different stages of pancreatic differentiation, standard normalization at the top and high CDC normalization with z-scores at the bottom. And what we can see is certain 3D interactions beginning to get set up that influence the promoter of this important diabetes gene PDX1. Okay, so we would like to understand how at different stages of differentiation, this enhancer rewiring influences expression of the target gene. Okay, so now I'm going to get to the machine learning part and tell you about a model that we've developed called GraphReg, where we're using graph neural networks to infer gene regulatory models. Okay, and the idea is we're going to use high C, or we're actually going to use a variant called high chip to encode 3D interactions, regulatory interactions as a graph. And we're going to propagate information along these edges in the graph via graph neural networks. Okay, so we think of the linear genome as a set of bins. The bins are connected by edges that we get from high chip data. The input features are going to be either 1D epigenomic data or DNA sequence, and the output is going to be gene expression. Okay, so the expression output of each bin. Okay, so we have two models, one that uses epigenome based data and one that uses DNA sequence. So I'm going to talk about the epigenome based GraphReg first. Okay, with this model, what we do is the inputs are chromatin accessibility and a minimal set of histone modifications. We actually just use one promoter mark and one enhancer activity mark. Okay, we go through a few CNN layers, and this helps us learn local features of the chromatin. And then we pass through several graph attention network layers. And this allows us to pass information between enhancers and promoters. And then we predict the promoter output as measured by CAGE-seq, okay, which is a tag-based protocol that maps promoter activity, the output at specific transcription start sites. Okay, so roughly speaking, this model predicts gene expression from the activity and connectivity of regulatory elements. This model is also cell type agnostic in the sense that if you train in one cell type, you can go to a new cell type, and as long as you have inputs, 1D and 3D inputs for the new cell type, you could predict expression there. All right, just a few more data. Information details on the model. We're using DNase, we're using HCK4 trimethyl and H3K27 acetyl, and a finer binning for the histone marks. But by the time we get to the graph attention network, we're at a 5 kb binning. Okay, so we are predicting at this bin, this resolution, the CAGE-seq, okay? And we're making a prediction on fairly large genomic regions of about 2 megabases. Okay, how well can we do? We actually can do quite well, all right? So this is true signal versus predicted signal on held-out chromosomes in mouse ES cells, and you can see a nice correlation. Also, if you train the model in different cell types, and then again on held-out chromosomes, try to use these models to predict fold change, again, the fold change looks good. Here, the color, the darker color means that you are looking at promoters with more high chip edges. Okay, so they have more complicated regulation. Okay. The other model, the sequence-based model, we start with 6 megabases of DNA sequence, one hot encoded. We pass through a few CNN layers again to learn local features, so now sequence motifs. And then we pass through dilated convolutional neural network layers in the bottom path to predict accessibility and histone marks. Okay, there's another path through the model where we take the output of this CNN, these motifs, and we pass to the graph attention network and try to predict CAGE-seq. Okay, so now we're using DNA sequence together with 3D connectivity to predict both gene expression and to predict the 1D epigenomic data as an auxiliary task. Okay, this is definitely a cell type specific model in that you're learning sequence information that is specific to the cell type that you train in. Okay, and just again, a few details on the model. The bottom path through the model is very similar to the dilated CNN model called Basenji, developed by David Kelly. Okay, so these sequence models that have been used in regulatory genomics. The novel part is the top path that uses the graph attention network. Okay, and again, the sequence model, it's a harder task, but we can evaluate on held-out chromosomes and look at true expression versus predicted expression, or fold change versus predicted fold change. And again, it's a reasonable correlation. Okay, and finally, I'm passing through this quickly, but if you compare the prediction of either the epigenome or sequence-based model to the corresponding CNN model, okay, so CNNs are widely used now in regulatory genomics. They're not incorporating 3D information. The GraphReg models do better, okay? And in particular, they are more, they give higher accuracy for prediction of gene expression when you start restricting to genes that are expressed and genes that have more complex regulation, more high chip edges, okay? However, unlike in a lot of machine learning, the prediction performance per se is not the point. We're doing this because we want to understand how gene regulation works and how individual genes are regulated. We want to interpret the model. So what we can do is use feature attribution to predict what are the functional enhancers for a specific gene. Okay, so what we're doing here, we look at a specific output, so expression of the gene DHPS, and for all the input features, we can use a method called DeepSHAP that identifies which bins of that feature contribute most to prediction. And we can sum up these contributions and get a track like this, which tells us which positions along the genome the model thinks are important for predicting this gene. Okay, and those, that feature attribution approach gives us a prediction of where we think the functional enhancers are. Okay, there's data to evaluate this now. This is thanks to CRISPR-I-based enhancer screening strategies. For example, CRISPR-I flowfish from the NGRIDES lab. Okay, so this data, what they're doing is they're taking a particular gene of interest, and they have a set of candidate enhancers, and they do a pooled CRISPR-I screen with a readout using RNA fish for the gene of interest. And they can, through sorting and sequencing, estimate the impact of perturbing individual enhancers on the expression, the fold change of the target gene. Okay, and in the same paper, they have a model for predicting functional enhancers called the activity by contact model. This is a score using accessibility, acetylation, and high-C contacts. Okay, so how do we compare with the ABC score or with standard CNN models? What we can show is that the graph regulatory models from GraphReg outperform these other approaches. Okay, so this is an evaluation on flowfish data and K562 cells. This is the performance by area under the precision recall curve over a set of genes. In green is ABC. Orange are the different GraphReg models. Blue are the corresponding CNN models. Okay, and so what you can see is that we have higher performance for determining functional enhancers based on flowfish data, okay, with the GraphReg models in orange versus CNN models or ABC, okay? And just a final slide to explain why GraphReg can outperform CNNs for this task. So what I'm showing is the MYC locus. MYC is an important gene, it's an oncogene. It's known to have very distal enhancers. Okay, so the promoter is here, and I'm showing the high chip data. And the question is, can these different models through feature attribution, can we figure out whether they're able to detect these distal enhancers? And what you can see is both the genome model based models and the sequence-based models, GraphReg can find these distal enhancers, whereas the CNNs can't. So 1D CNNs, the dilated CNNs, in principle have a wide receptor field, but actually the feature attribution shows they're only learning information very proximal to the promoter. Okay, they can't access this distal information, and that's the power of this approach. Okay, so I showed you some new work using graph neural networks to predict gene expression across large genomic regions using both 3D interaction data and 1D epigenomic data, or using DNA sequence with 1D epigenomic prediction as an auxiliary task. And I showed that the GraphReg models outperform the baseline CNN models for gene expression prediction. More importantly, we can use feature attribution to predict functional enhancers for genes, and this outperforms existing models as well as the ABC score for identifying functional enhancer elements. Okay, and I think the big picture here is that there have been rapid developments in machine learning modeling, in epigenomics, in 3D genomics, and also in screening approaches based on CRISPR editing. And this has all enabled advances in modeling gene regulation and deciphering regulation. Okay, with that, I want to just thank all the people in my lab who did the work. Alireza Karbala-Gyare is the postdoc who developed GraphReg. Marve Sahin developed the HiCDC Plus package with help from Wilfred Wong. And in particular, I want to thank my collaborators, Effie Apostolou and Danwei Huangfu, as well as funding sources.
Okay, great. Thanks very much, Christina. So let's go to a couple of questions just for this speaker before we move to the general discussion. I'll start with the first one by Jens Lichtenberg. How difficult would it be to infer the deconnectivity when assuming the other features such as epigenomic data and expression data are given or well known? And Christina, maybe we can, just for the benefit of the audience, explain what is meant by the deconnectivity.
The connectivity, like the high C contact
map? Yeah, I'm guessing what they mean is the high C connectivity from this question.
Right. So that's a great question. So GraphReg uses high C information, the high chip information in the model to predict gene regulation. Another problem would be trying to use 1D epigenomic data to predict the contact matrix to predict
exactly.
Contact. And we and others are working on it. We are working on it in the context of using both bulk 1D epigenomic data, as well as using single cell attack data, using different deep learning approaches. There are existing approaches that go from DNA sequence to the contact matrix, training in a specific cell type, but one doesn't expect those models to generalize to a new cell type.
Let's see another question from Antonios Liutsas, which starts off with saying, great work, very exciting. A lot of the work that you're presenting is on population models. And I'm wondering if your ML models would benefit by using single cell data in addition.
It's where we're going, so I don't, I'm not quite ready to put it into the workshop slides, but we and many other people are looking at multi-omic data where you have single cell attack and single cell RNA-seq in the same cells. And the cage-seq that we're using looks very much like the signal you get when you summarize single cell data, especially certain kinds of assays over cells, over clusters. So we are definitely heading in that direction, as are many other people.
Great.
And actually, maybe I'll ask you a question on my own. In natural language processing, there was kind of a phase transition when transformer models came in, and you see kind of the rise of BERT and GPT-2 and 3. Kind of your thoughts on whether transformers would be useful in this domain, which at least to me looks a little similar.
Yes, I was actually just reading a paper yesterday, which is from Calico and DeepMind, David Kelly's. So it's a preprint, and our GraphReg is also a preprint. So they're both available in bioRxiv, and they're using a transformer architecture. So it's a different attention-based mechanism. They're not using 3D structure, but they can, through an attention mechanism, get some longer range interaction. Not quite the 2 megabases that we are getting, but they can kind of learn information out to about 100 kb, which is impressive.
Okay, great. So why don't we go ahead and get all the speakers of the session up here, and we'll start the general discussion period.
And maybe while we're waiting for some new questions to roll in, I have a question again for Jan Peng. It was kind of amazing to me how so much of the advance in protein structure came from methods that are based on information theory or evolution, rather than from a biophysical approach. Your thoughts on why that is, and whether or not we'll actually get to a place where the biophysical approaches kind of catch up to the more information theoretic approaches.
Yeah, sure. So I think first, the biophysical based approach has their limits, right? Our current understanding on, for example, man-body interaction and the exact formulation of how these potentials should be formulated are not complete. Where we have although there are a lot of progress in the field, we don't have a very much understanding on these, and also the biophysics-based approach also suffer from what we have seen in optimization. There are a lot of issues in optimization. Those energy functions are highly rugged. There are, in the landscape, people have visualized that there are a lot of local minimum, and those energy functions are really, really hard to optimize. So I think that's the main bottleneck of applying those approaches in large scale, and of course the computational costs of those biophysical based approaches are really, really demanding, and we need a lot of supercomputers to perform a lot of simulations and to get some reasonable folding or simulations. So I think the future direction might be as you said, the information theoretical based or machine learning based plus physics-based approaches. For example, in our approaches and also many other groups, especially also in DeepMind's approach, we're not just only use the deep learning approach. For example, for protein structure prediction, protein folding, what the machine learning is giving us is just the initial structure. We got this scaffold to be right. We know, for example, this particular residue should be placed in the right direction, but the details are not good. So usually what we do is that after we obtain the initial structure, we use a force field or some of these biophysical based approach to optimize those atom detail, atomic details, a lot of sidechain packing, orientation, etc. Because if we don't have that, the structures globally they look correct, but if you look at the detail, they are not useful at all. So that's why people now use these kind of different approaches and put them together. And machine learning, I would say, is very good at identifying a good starting point. After we find this good starting point, and then the biophysical model will be very helpful, useful to optimize the detail and get them folded into the right structure.
So a question just came in that I have to say I've been wondering during a number of these talks, and so I think it's a good general question for everyone. How much data do you think is needed for genomic deep learning? And I would say you're welcome to comment more generally, but start with your own research. And let's start with Christina Leslie, because Christina, during your talk, I was wondering, you're running this thing, obviously you're scanning it along the genome from chromosome to chromosome, and you mentioned you can use one chromosome as a holdout to validate the model and others. But is that just one individual? Presumably you have multiple individuals, and so we have these sort of at least two dimensions, number of nucleotides by number of samples. Is that how I should think about this? And how much data do I need in both of those dimensions?
So let me point out the data I'm showing you is training on a single cell type. So, and it's, ENCODE cell line data or mouse CS cell line. And a lot of work so far has been at that level, or, maybe you do multitask where you're predicting all of ENCODE, right? But fundamentally it's predicting it from the reference genome. And as we move forward, it's not magic. We're going to need to incorporate genetic variation into the models if we really want to learn what is the function of non-coding genetic variation on gene expression. So, and we have some strategies. Other people have strategies as well. But that's the next step.
But for instance, do you have any estimation of how things will improve as more data are added, as more cell lines, for instance, are added to the repository? Is that going to help?
I don't know. So I actually think instead of cell lines, I think we need primary cells looking at differentiation stages, like in our 4DN project. We want to model something that is relevant both to cell identity and the biology of the fully differentiated cell and the biology of disease, right? So I think we should be training in more relevant contexts. In terms of learning, so the epigenome-based model that I talked about, where from a few marks and the connectivity, we're learning something fairly modest about how contributions of distal elements and the state, helps us predict the level of expression output. And so it's not surprising in a sense that does well. We have good information that can generalize to a new cell type pretty well. But it doesn't get us all the way to what does this, the minor allele change in regulation of an important developmental gene.
So Sarah, how would you answer the question about how much data are you using, and what are your views in general on obtaining more data and how it would impact the machine learning you're doing?
Yeah, I think it's a different application than what Christina was talking about, but we do use the entire genome, but we would do that for multiple individuals, usually say 200 haplotypes, so 100 individuals. And I think it hasn't been explored enough, how many we actually need to do well. Could we get away with fewer? I think there's kind of some interesting questions there. And I think it depends on what you want to answer. For natural selection, you may need more individuals to really see that pattern. For things like ancient population size changes and migrations and admixtures, you may need only fewer individuals. But I think in terms of, I'm also sort of, chopping up the genome into regions, and I'm trying to look at feeding each of those regions in almost as an image, right, to the GAN framework. So how many images do I need? I mean, I would say a lower bound would be something like 10,000. I mean, just to throw it out there. So that kind of there's a trade-off between taking more SNPs per region and having fewer regions versus taking fewer SNPs per region and having more regions, right? So I think there's a lot of exploration that needs to be done there, but we're kind of, for GAN, we're limited in just the length of the genome, right? Whereas for some supervised machine learning method where you're using simulated data to train, there's no limit to the amount of training data you have, right? It's just, is it actually good quality training data?
Great, thanks. John, did you want to comment as well?
Yeah, I want to just add one comment that although for a lot of problems, like we don't have, for example, we don't have a lot of cell lines, we don't have a lot of samples, we don't have too many genomes, or we don't have too many proteins to train all these models. But essentially, you think of all these like modern deep learning models, they're trying to capture these local patterns from very bottom and organize in a way that to come up together with more complex organized patterns. And a lot of these, if you think of these local filters or local feature extractors at a very lower layer in these models, we actually get a lot of data, right? One, we have 3 billion of base pairs in the genome. Each of this region can be seen as individual data points. Of course, depends on what kind of task you want to do. Like for example, for proteins, like we don't have a lot of proteins solved, although much more than we see in the past. But essentially what we're trying to do is find all these interacting motifs. Those are local interactions. So essentially I would say why these deep learning models work is that although we don't have a large sheer number of how many samples we can get, but within each sample, we actually can construct a lot of useful information to create the data points. That's what I want to comment on.
Excellent. So, actually, Anshul Kandaji asked a good question in the chat here, about what you could say in general for stability or instability deep learning models, if not, in fact, all classes of ML models. Maybe we'll start with Christina. Would love to hear your thoughts on that.
Yeah, so we're, moving into this deep learning space because I think the models are very exciting and flexible, and they're allowing us to do things that we couldn't do with the previous generation of machine learning models. But I think Anshul is exactly right that it can be scary how unstable they are, and you do have to use good machine learning practice, right? And you have to use ensembles. You have to be very careful not to fool yourself, as with all machine learning, that you're really generalizing. So I agree. It is it is something that we all have to be cautious about.
Well, and just to push on it a little bit, Christina, how does the stability change as you start to add even more parameters or more complexity? There's a certain kind of theoretical train of thought that's arising around over-parameterization actually being kind of useful rather than detrimental. So as our models get more complex, will they get more stable or less?
I don't know that we can make a general statement, but yes, we've also seen these papers and also empirically the idea that you want to over parameterize and maybe it makes your gradients better, right? And more ways to kind of approach a solution. It's very interesting. I think it's sort of a lesson for learning theory that the algorithm is part of part of understanding whether whether you overfit.
Excellent. Maybe we'll go on to a few other questions just to divide it up. Maybe Stephanie, you could say a little bit about what are the right inductive biases to use for genomics. And again, just because it's a general audience, maybe you can start by saying a little bit about what is meant by the term inductive bias. Sorry, I meant to say Sarah. I meant to say Sarah. Sorry about that.
Okay. Yeah, I think it also maybe depends on the application a little bit. I think there's sort of, I guess, biases in the data itself. And then there's also biases in terms of the methods that you choose, right? And that might be your own inductive bias about what you think will work better. I think in population genetics, this kind of manifests itself as choosing models we think are realistic in terms of evolutionary models, say like clean splits between populations, and we oversimplify in that case, and I think that can lead to some inductive biases. I think they would be a little bit different in each field, but I think what I would like to see is actually methods that allow you to move between models. So often we fix the model and we fit it, right? In terms of, say, two population model with a clean split and no admixture census split. So what could we do to actually allow the method to explore models, and so that we are not constraining ourselves in that way? I think that would be super interesting, and would kind of get us away and maybe explore solutions that we didn't think were really feasible, but actually happened over evolutionary history.
So one discussion point that keeps coming up, I think it came up in Sarah's Q&A, that I think is good to discuss as all of us, is this role of simulated versus real data, and in particular the role of simulated data in what all of us do, who are up here on the virtual stage. On the one hand, simulated data can produce virtually an infinite number of training samples, and has lots of other benefits. On the other hand, if you're not careful, you've stopped learning about human biology at some point, and you're learning about how this machine learning algorithm works, which of course is less interesting. So Sarah, why don't we leave you on the stage, or on the floor, so to speak, and maybe you can comment, just in general, how do you think about simulated data, and when are the appropriate uses versus when are the inappropriate uses?
Yeah, it's a great question, because I think that in population genetics, we've relied on simulations, really heavily, perhaps, more than most fields. And that's really because we can't really go back, in a sense, in the past and find this evolutionary ground truth. So if we wanted to infer historical mutation rates or recombination rates or population size changes, we don't have the ground truth. So we are forced to rely on simulated data for anything that's sort of supervised, broadly defined. However, I would say it's not as bad as it sounds, because we do have actually very good evolutionary models. We know how to do the forward process. We just don't know how to infer, or that's the goal of population genetics, sort of to infer going back in time what actually happened. So I think that it still it still hampers us a bit, because we only can simulate some things that we think are real. But I think we do have very good evolutionary models. I guess there's two exceptions to that. One is ancient DNA. So if we had really good ancient DNA, we could actually have some ground truth. However, it wouldn't be nearly enough to train any, okay, that's what I think right now. It wouldn't be nearly enough to train any machine learning model. And two is experimental evolution. So you can do this in yeast, or you could do this in, some other type of fast reproducing species, but I don't think you're going to be able to do experimental evolution for human, right? So I think that's where we will maybe always rely a little bit on simulations to test our model, to validate our model. Does it produce reasonable things in simulations? Maybe we can sort of triangulate with different models and say, okay, if all these models were tested in simulations with different caveats and things, and they all produce the same result on real data, maybe we start to feel confident this is a real result. But without external validation techniques, we won't exactly know what happened, thousands of years in the past. I think there's a power of GANs, that's what I'm interested in right now, to help us make better simulated data, more realistic simulated data. I think we should think about like sort of underlying structure in the data, clustering and things, visualization that helps us learn more about the data in an unsupervised fashion. But I think we're always going to rely a little bit on simulations.
John or Christina, did either one of you want to comment?
I mean, I think it's not clear how we do data augmentation in the genomic setting, right? Whereas in the image setting, there's lots of strategies to augment your training data. So, augmentation in a way that doesn't bias badly.
I would say the simulation data are useful to validate hypotheses. So basically, you want to develop method, you want the method to capture certain like inductive bias you believe to be true for this particular biological problem. Then you like to simulate the data so that the data have the property you wanted it to have, and then check the model is useful or not to detect this kind of thing. So essentially, I think all of this all these questions and the previous question are all related in a sense that what do we really want to do to use this machine learning or deep learning approach for? Whether we just want them to improve prediction, that's okay. And for example, we want to predict the protein structure. Sometimes we don't need to worry about how these structures are generated as long as it's useful, right? For drug discovery, etc. And for many other fields, like especially I know in the system biology and regulatory genomics, people want to find the tools that are able to identify certain knowledge, right? Certain kind of interactions. We don't know whether those sometimes these interactions are very complex. So we don't know whether the method is able to mine such a relationship. So we generate the data set so that we can learn these kind of patterns. So this also comes back to their original question: how do we actually use a deep learning in the right way for different problems? I would say for many problems in genomics, the accuracy is secondary, right? We don't really want to push the accuracy to be very high. We want the model to be insightful. We can use the model to discover a lot of new knowledge, a lot of new interaction, new insights that will be better for us to better understand biology. So I think all of these are related questions, and it's all about how we actually design the right methodology to interrogate the data, to identify what our purposes are, whether we want to make the accurate prediction for some downstream analysis or our downstream applications, or we want to find a new biology and better understand the biological system.
And thanks for the segue to interpretation or explainability. So one thing I was going to ask you, John, is both Sarah and Christina got questions about interpretability of their models. You didn't so much. So I was going to ask you, how do you think about interpretability or interpreting the models, say you're building for structural proteomics and protein folding?
Yes, so we actually consider that very much. So for example, when we build a model to predict a protein structure or protein contact structure or interactions, we actually visualize. So there are a lot of approaches developed in the field to visualize activations of certain layers in your neural network. So we can, for example, you can essentially, what you want to do is to take the gradient with respect to the input, right? And or you just across different type of data, you visualize which neurons are activated. In this way, we can actually go backwards to the original sequence or structure to identify whether there's some relationship between, for example, two beta strands or two alpha helical structure interacting with each other. So we also do that, check that all the time. But instead, I would say the goal of this problem we study is kind of different from other genomics problems. We want to push the accuracy to be to a regime that we can use a prediction. And but for the other field, like Trey, you have done a lot of work in this field. We all understand that we want to figure out what the model actually learns. Basically, essentially, I would say at the end of the day, what you actually learn that can be transferred, can be generalized, are the very simple rules, right? Some biological rules. The rules are noisy rules. We cannot easy to learn using very simple models. So that's why we need deep learning. But finally, we need to distill this kind of knowledge from the model. One way is to using this interpretable approaches like a postal analysis, right? After you build a model, you want to analyze what you have learned. Another way is like what you did and others did is you use this inductive bias, build the model in a way that the model already incorporated necessary knowledge of biological information into the model, so that you can visualize the model and make conclusions and find a new discoveries, right? So I think those are two different approaches people widely use in the field. I don't know which one is better. I think both have their own virtues, and but I think this will be I think it will be a very important research area in the future. Because in the end, we want to discover new biology, want to better understand the biological system.
Yeah, I would agree. And what I presented, it's very important to be able to predict gene expression because you want to learn the mapping from the epigenome or the genome genetics to expression. But actually, expression is the easiest thing to measure of all the things that
right.
The model, right?
That's right.
So if you just wanted to predict, you could just measure it.
Just measure it, exactly.
I have a question for the panelists that's a little bit more related to kind of training. One of the things you all three have highlighted is the challenges of actually getting your models trained. And there's a fair amount of kind of knob turning that has to happen. And that's the kind of thing that really is the domain of experts who really learned in an environment how to do it. But now imagine you're a lab that hasn't yet done a lot of work in machine learning, and you want to start being able to build your own models and being able to apply them to the kind of question of interest. What's the best way to get up to speed? Is it by finding a collaborator, recruiting a postdoc that knows how to do it, or just blood, sweat, and tears? Maybe Christina, you can start.
I'm a big believer in bringing in postdocs who are well trained in the area and getting them excited about biology, right? And having a training environment where you can say, look, you have exciting methods, but we have the real problems, right? And we can put this methodology to use in a really impactful and meaningful and biologically sensible way. So yeah, so you have to lure them away from Google and Facebook and get them in your lab.
So you mean there are some people who'd rather work on cancer than selling ads?
Yeah, this is all all we have is, you know, come cure cancer. We can't offer the salaries, or, you know, that we can, there is a mission.
You know, I see we're almost at time, so maybe actually we'll just end on that note. I thought this was just a wonderful session. I'd really like to thank all of the speakers. And there were many questions, really great questions that we didn't get to today, but I thought this was a great discussion.
Thanks, everybody. That was fantastic.
Thank you.
1, 2, 3, 4, 5.
Hi, Dave. Are you on?
I'm here.
Hey, how are you?
Good. How about you?
Not too bad. I had to step away and then dash across campus to make it. So just about right. Can you see I did the NHGRI background. You can see it. Okay.
Let's see. Yeah, it looks okay. I guess mine looks okay. At least.
Okay. Hello to our speakers. I see Pamela and Varun and
Downton Char.
Oh, great. We have everybody.
Hey, how's it going?
Hi, how are you? Okay.
Do we need to do a new orientation session, or everybody knows what happened?
An orientation on which part?
Well, it's just what's going to happen now, because I'm not sure if everybody in session 2 already attended the orientation at the beginning of the day.
Valentina, just keep in mind, Valentina, that we are still live, so
yeah,
just
we
kind
are.
of aware.
Okay.
You can orient or you can chat a little bit. I think it's fine to talk a little bit to people. This is sort of like you're just getting your preview as if you were up by the lectern in between sessions, but I did want to chime in just to make sure everyone knows we are live. Everything you've been doing so far is good. It's always a good warning.
I'll be quiet.
Especially before I start saying what I really think about Dave. I'm just kidding, Dave.
Thank you for reminding us, Carolyn. That was helpful.
Yeah, thanks, Carolyn.
Amira and Dave, I just want to give you a heads up that on your screen, you'll see a 5-minute countdown timer. So you'll be aware of when to actually start.
Great, thank you.
Mm-hmm.
For the speakers on the from the last panel, and thank you so much for such a great panel. For an LC researcher, as long as we're sort of having our little conversation at the podium and others can overhear us, can you compare the complexity of the models you all are fitting to the complexity of something trying to inform clinical decisions? Are they similar, or do you have any sense of that? Or is it even a a reasonable question to ask?
So Dave, this is Trey. I'm not a speaker, but I'll chime in with an opinion.
Yeah, please, Trey. I appreciate it.
I think these models are not so far from clinical models after all. And I don't think we heard any direct clinical links today, but if you put them in a broader family of models, there are clinical inroads. And so I think, the model's more focused on understanding molecular effects like Christina's gene expression, or Jinpeng's structural biology models. These are all one step away from the clinic. Just the connection has to be made.
Yeah, I think I didn't ask my question very well. It's easy to see the connection to the clinic. What I'm wondering is how complicated are clinical machine learning models compared to the ones which seem awfully complicated that Christina and John are running?
At least in our own lab, they're really similar. The architecture.
Gotcha. Okay, great.
Yeah.
Once I get beyond 6 variables, I'm lost. So.
We're starting again at 3:10, is that correct?
Yes, that's correct.
Yeah. Good afternoon, everyone, and welcome to session 2 on ethical, legal, and social implications, or the ELSI of machine learning and genomics. I'm Dave Kaufman. I'm a program director at NHGRI in our division of genomics and society, and Emer, my co-moderator.
Hi, everybody. Yes, I am Emer Kenny. I'm a professor of genetics and medicine at the ICANN School of Medicine in New York City.
We're really glad to have you here, and we want to get going since there's a lot to hear and talk about. Our first speaker is Pamela Sankar. She's an associate professor in the Department of Medical Ethics and Health Policy at the University of Pennsylvania. And we'll get started with her talk.
Good afternoon. I'm Pamela Sankar from the University of Pennsylvania in the Department of Medical Ethics and Health Policy, and I am going to talk this afternoon about ethical issues associated with machine learning. And I'm very pleased to be part of this workshop and very much looking forward to the discussion that we have afterwards. Today, the approach or the kinds of information I'm going to talk about machine learning is a little bit different than the way much of the ethical literature has dealt with machine learning. A lot of that work has focused on issues that are embedded in the technology. So transparency, bias associated with sampling, explainability, accountability. A lot of the other work in ethics about machine learning deals then with more standard medical ethics concerns. And what I want to do today is sort of move a little bit past that, and we'll see as I go along what I mean by that. We've heard a lot about machine learning, maybe a little bit less now than people are accustomed to. I think they declared 2018, somebody declared the year of machine learning in healthcare. But it is the case that we still see a lot of declarations about the importance of machine learning and the claims that it is the technology that will transform healthcare by reducing costs and at the same time improving quality. People have begun to question whether this might really happen, and so there are people who, as the second quote there suggests, asking whether machine learning is a giant with feet of clay. I think that studying the progress of machine learning is difficult. I know we have found it difficult because a lot of the work is proprietary. So you might learn about a project going on, but it's hard to keep track of it. Other projects are done, say at universities, are done under QI or QA. And so very often the results are not published outside of a particular institution. That said, it seems clear that progress has been made in areas like drug discovery and oncology, whereas progress into clinical settings of machine learning has been slow. As these authors observe, examples of fully integrated machine learning models that drive clinical care are rare. This development, this development allows clinicians with limited machine learning knowledge to apply ML models to their data sets, and yet when they looked, they were not successful in identifying any applications in translational medicine. And the final one said, given this impressive array of studies, it is perhaps surprising that real-world deployments of machine learning algorithms in clinical practice are rare. So one question that we were interested in figuring out is why the clinical applications were slow. I'm working on a project right now with Dr. Mildred Cho at Stanford, funded by NHGRI, by ELSI. And in it, we are interviewing people doing machine learning development for applications in healthcare settings. And I'm going to review some quotes from these interviews that address the question of why clinical applications might be slower than applications elsewhere. One reason is that the people doing a lot of this work are unfamiliar with the healthcare setting. They have not worked, many of them have not worked in healthcare setting before. And they are challenged by what they encounter there in a very specific way. This quote. One thing to ask about an algorithm being developed is, and it can be really scary to ask, is but like kind of the worst case scenario. And I think that's a lot of the problem with healthcare is, you know, the worst case is that someone loses their life, you know, I mean, or it gets worse even. It's just versus, you know, someone's Uber didn't show up. It's a different worst case scenario. Often they're comparing their experience to working in the financial sector, this person comparing it to Uber. Second quote, and so many of these AI groups just go into this big data like it's data and don't understand the consequences. I mean, there's not much consequences in telling people to buy the wrong book, but there's a lot of consequences in telling people the wrong thing in healthcare. Here's another similar sentiment. I think there are some issues there, but I think the disconnect that I've seen is in the ethos of how computer science has turned into a business. It has been built around this notion of tolerance to failure. The cost of doing it is cheap, so we can fail and move on and do another one. That is antithetical to medicine. You're not allowed to fail with people. That's not an acceptable ethos. What this person was talking about was having worked in the app domain, where obviously when you put an app out on the market, you want it to be relatively complete. But if it isn't complete, you get a lot of feedback and you can fix it. And so that's the mentality. You can always fix things. One of the most consistent themes that people talked about was problems with electronic health records, figuring out how to deal with those. And on the flip side of it, when they do send us labs, and they here is physicians, labs and other clinical data, usually in extracts from an EMR system, then that half of the battle is trying to make sense of that really, really crazy data to make it fit alongside the algorithms we've already built previously. The second quote. But they're interacting with like the user interface end of Epic, and they, the physicians, they'll say like, well, I just used the social history tab to code that. But then we're looking at massive data tables in the back end and trying to figure out where all the stuff that they charted ended up. The problems with medical records, as I said, is the most common one cited, and it really speaks to a broad issue, I think, in machine learning. Here's a quote from one of our respondents. We need clinicians caring for patients, not entering data into systems, which for the most part do nothing with it. But this is a completely backwards approach, right? What we're basically doing, instead of designing a system that says, what do we need? Let's go make sure we have that data. We're saying, okay, why don't you, doctor, nurse, enter a ton of stuff, of which probably the majority is not very useful from an ML standpoint. And this is an important, interesting point, I think, because it was a theme that I heard several times talked about in January 2020 at the Precision Medicine World Congress out in Santa Clara, California, mentioned by people who were very prominent in the field, that on panels, that nobody was actually saying that they were going to jettison the EHR, let's be clear, but that it was coming up as people started to think we need to think about whether or not it would just be easier to create new data systems de novo. Another set of issues that are respondents didn't bring up really, but I think is important to recognize, is that there is a sense from internal to the information sciences that the research that they're doing is not rigorous enough. And these are two examples. A rigorous review of articles reporting studies of deep learning performance against healthcare professionals in detecting diseases from medical imaging reported that of 82 studies that qualified for detailed analysis, only 4 provided physicians with conditions that mirrored real clinical environments. So that might be a problem when you think of all of the studies that have come out recently saying that machine learning outperforms physicians at various tasks. You might want to look carefully at how they created the conditions for the physicians and the machine learning. And then this second one is an essay on the pitfalls in machine learning research, which opened with the claim that research progress was hindered by reliance on ad hoc design, process, poor data hygiene, and a lack of statistical rigor in model evaluation. So all of these things suggest reasons for factors that are slowing down progress in machine learning in the clinical setting. The machine learning developers were unfamiliar with healthcare challenges. They were animated by a different ethos and bewildered by the complexity of and the multifunctionality of medical records. What we might say when we look at those explanations, and I meant to stay on that slide, is that this is evidence of a young field and that these things will change because people will become more familiar with what they're doing. And there's a very strong emphasis in working in teams. And I think that is also something that will amend some of these issues. But there also is something more here that I think is interesting to look at, because the only salient feature is not inexperience. It's how did this experience get so close to healthcare? How did people whose expertise is in getting you connected to your Uber end up designing algorithms to detect risk for diabetes? It's important to consider, one way to explain this is to consider the history of machine learning. And machine learning was developed as what we would call a general tool. Machine learning was created to be a general tool designed for general purposes in a general context. Machine learning emerged from efforts to create a capacity for computers to learn from their own operations using data being processed from streams of information fed back by satellites to credit scores, As it evolved, success was judged in part by how readily it could be adapted across work settings. Thus, machine learning developers who know very little about healthcare got so close to healthcare because it never occurred to them, or apparently anyone else, that they shouldn't or couldn't be doing this. Moving from finance to healthcare would be no more difficult than it had been to move from finance to marketing. The hammer would find its nails, data would be wrangled, and algorithms trained. They look only or primarily at the tool, not at the context, and they're unprepared for the complexity that healthcare has provided. Interestingly, getting to the ethics more, there's a parallel in this, in the ethics literature, and that is an overfocus on the technology of machine learning, on what seems to be imminent in the technology. So here are some quotes that demonstrate that. Thus, the machine learning algorithm might face difficulties at providing a reliable medical diagnosis for a patient coming from Eastern Asia. Moreover, studies indicate that image recognition software is prone to develop biases. Next one. If algorithms trained on data sets with these characteristics are adopted in healthcare, they have the potential to exacerbate health disparities. And finally, standard machine learning can acquire stereotyped biases from textual data. And in all of these, the machine learning is reified. The people are removed. The solution proposed in these articles, the most common solution proposed in these articles, is to pay careful attention to the samples used in test data sets. And that, of course, is the right response. The question that I want to ask is, what does this kind of a response imply? What does it say in terms of how to pay attention to health disparities in the context of machine learning? The focus on the tool as the source of the problem suggests that problems can be solved by modifying the tool. This is what much of the ethics literature focuses on. Effort is directed toward blunting harm, to improving transparency or explainability, mitigating bias by paying attention to sampling, all of which is necessary, but not sufficient. Because efforts to improve population health without worsening health disparities have to go beyond minimizing harms. They have to seek to maximize benefits. Specifically, they have to seek to promote health equity. The ethical challenge then posed by machine learning in healthcare is not to let interest in machine learning obscure our commitment to ameliorating health disparities, which may or may not be achieved with the help of machine learning, but in any case would require more than careful sampling. That's it. Thank you.
Thank you so much, Pamela. I really appreciate that talk. And maybe we can start with a couple of questions from the board. Someone asks, and I think it's a good question. I think it's, I think something that you point out is the need for kind of multidisciplinary understanding. And so someone asked, do you think certifying machine learning engineers in related medical domains by the medical board would help to increase more clinical applications?
Say the last thing again, certifying by a medical board?
Yep, certifying a machine learning engineer in related medical domains, would that help to increase more clinical applications?
Sure.
Training.
That's a possibility. That's a possibility. I do think that building off the natural or the way it has evolved, which is that there's a lot of team work. And if the integration of domain expertise, of medical and clinical expertise is more routine, more available, I think that is a way to mitigate this problem. It's interesting that they have the idea of board certifying somebody. That's possible. I mean, one of the things that I was going to say, which is related, but in a different direction, is the idea that in terms of being able to assure or promote machine learning that addresses health disparities in the sense of promoting equity, not just of reducing risk, but of promoting equity, which is not the same thing as, I mean, it's good to reduce risk, but promoting equity really is a broader, a more robust idea. That what I was going to suggest was that they start requiring, in terms of FDA approval, that there be expertise, demonstrated expertise in an organization, in a team about health disparities. But they could certainly also do what the speaker suggested.
Thanks. Another question is, how do you think the landscape of ML and AI in healthcare might be affected by the entry of big tech, which already manages so much of the data in the healthcare field?
Big tech has essentially created the current landscape of machine learning. It has so profoundly shaped what's going on, because even if certainly there's a lot of work going on in academia, but when you leave academia, and even with academia, many of the projects that are being organized now are through partnerships, many of them with venture capital. And the relationship between venture capital and big tech is very intimate, and it's not like it's two spheres. It's venture capital incubating ideas that then big tech takes over. They're very closely related, and it makes for a very disrupted field. It's very hard to require certain kinds of regulation and certain kinds of planning, because so much of the work takes place in these isolated, small, isolated from the perspective of researchers. I'm not saying they're isolated, but in these small startup organizations. So big tech has shaped what we are now seeing, and they've been there for 4 or 5 years at least, maybe longer, I don't really know, but they have shaped the field very strongly. In fact, it'd be interesting in the discussion to hear from people who are working in academic settings or at the VA, places like that, where their organizations have made contracts already with Apple, Microsoft, Google, Facebook, et cetera. It's created the landscape as we now encounter it.
We talked about this a bit earlier, but do you have a sense that there's resistance from clinicians, and does that have any bearing on the pace? And I guess with respect to the pace of implementation, given some of the difficulties, I wonder if you think the slow pace is appropriate. But also, do you think that sort of clinicians' concerns about machine learning applications, either replacing them or just not trusting what they have to say, is that a barrier you're already seeing, or is that something that we're anticipating?
Certainly in isolated situations. I mean, I don't pretend to have a grasp on all of the projects going on. That's actually one of the things that's sort of hard about studying this field. It's hard to get details. But certainly there are examples of physicians that are resisting, but there's also many, many examples of physicians that are participating and promoting it. I think that some physicians accurately sense that the machine learning they're being asked to implement is not designed properly for their context. And that's an important thing. I mean, one thing I want to make clear is that the people that we're talking to now, almost all of them are not affiliated with academic centers. And so they're probably unlike many of the people in the audience. These are people who really come out of machine learning. And a lot of the work that they've developed, the algorithms that they've developed not entirely on their own. I mean, they have domain expertise, they have physicians, but they're working in a way separate from the healthcare context that it suggests a reason that it explains why they often come up with applications that physicians resist. But I'm not sure that the physicians are resisting it so much because of the sort of traditional predictions that, oh, it's going to replace me, so I don't like it. I think there's distrust, but I think the distrust is based on experience, not on belief.
Thanks. And maybe a follow-up to that. Shub, I see your question. I kind of want to hold it for a bit later because I think more people are going to talk about that. But a question that I got from some of the panelists, do you think that I think touches on that is whether you think increasing the interpretability of the models is going to increase trust in translating them into the clinic? But, a related question.
Sure. Yes,
Do
of course.
people need to,
I
an
think,
argument we have with genomics sometimes is that not all providers need to know everything about genomics. And one could say the same thing about an ML model, but does there need to be some level of understanding? Sorry to interrupt you.
oh no, that's okay. But it's it's a networking process, right? And so no, not every single practitioner needs to be able to interpret it, but those practitioners probably would like to know somebody who can interpret it. And so it's time, it's over time. It's like the more physicians become expert in this, the more physicians who understand it, there will emerge within the profession, within healthcare, a sense that, oh, well, these people do understand it. Because of course, there's many technologies people use all the time that they themselves cannot explain. But that doesn't mean that they don't trust them. But the trust is built through watching other people use it, having institutions approve it, having a record of good outcomes. So I think interpretability will help that inner core. Enhancing interpretability will help the inner core become more trusting, and they'll understand it better. And as the inner core becomes more trusting and understands it, then chances are, not to predict everything, but it will diffuse out. I mean, that's true of technology outside of healthcare. That's just a process. There's opinion leaders, and then it goes off into the broader group of people. And as long as there are no serious problems, well, there will be some somewhere, I'm sure. But I think that the general trend will rely on people's networks and people's acquaintances or the professional organizations they're associated with.
Thanks so much, Pamela. We'll bring you back for more questions during the broad discussion, but it's time to our next talk. Emer?
Great.
Okay, thank you.
Thank you, Pamela. So our next speaker is Varun Mothur, who is a research fellow at the AI Now Institute at the New York University, a fellow New Yorker. Off you go, Varun.
Hi, thanks so much for having me today. My name is Varun Mothur. I'm a technology fellow at the AI Now Institute, which is a multidisciplinary, interdisciplinary institute at NYU's Tannen School of Engineering, focused on really trying to understand the social impacts of artificial intelligence in systems. And today I want to be able to talk to you a little bit about our work and maybe how we're thinking about ethical systems can help inform some of the work you're all doing in machine learning and genomics. I'm coming to you live from Vancouver, from unceded Musqueam and Squamish territory. So I just wanted to start a little bit with sort of the provocation of exactly how we think about the social implications of these systems. And by doing so, I want to focus a little bit on two examples that came up with AI and healthcare in the last couple of months. On the left, what you're seeing is the headline from Vox on DeepMind's ability to create an AI model, which was a neural network that basically cracked sort of the benchmark at Cas14, which is trying to understand protein folding and predict how proteins would fold, given their sequence. And so that was a really interesting use case for AI systems that definitely sparked a lot of ideas around potential for AI in healthcare. But on the right, what you're seeing is a headline from months that just followed after, which was an AI or an algorithmic system used by Stanford's hospital network to distribute vaccines. And particularly the issue with that was the people that were prioritized in that system were not frontline care workers or resident physicians, but physicians that were senior and were probably not on the front lines or working from home. And so the question that really begs us from is, like, how do we define an algorithm, or how do we define sort of the ethical and social norms that govern these systems? And I think Christian Lum and Ruman Chowdhury both wrote a piece later on in MIT Tech Review stating that, maybe instead of focusing on the technical definitions of an algorithm, we focus on the harms, and centering the harms and the actual impacts that people have, regardless of whether or not the technical specificity or the complexity behind it differs greatly, but rather focusing on those harms is what really will allow us to elicit and understand sort of what those ethical implications ought to be. And I think that's profoundly important, especially in the world of genomics. This is a report that was created by Khadija Theraman and Michaela Pitkin at Data and Society, which I think is a really great report that outlines sort of the larger social impacts of sort of precision medicine and the larger development of medical AI systems, primarily focusing on historical bias, on the lack of data, specifically around people of color. But I highlight just one of the things that they mention is that the use of data specifically is tied really to inequalities in spaces like criminal justice and welfare and child services, primarily because they have a surveillance component to them. And so how does that actually translate in genomics? And it's something that might be interesting. I guess one of the citations that they actually use at the very bottom there relates to, eugenics. The idea that what are the scientific correlations that are being used to build, develop systems that are then impacting people in real time? And so how does that data get misused, or how does that data not align with the good of people? And so I think that's kind of where we as an institute are coming at it from. We're an institute that's focused on creating policy recommendations around around the use and the governing of these systems. And I think one of the recommendations that we authored, which was looking at how we can move beyond technical fixes to address the larger political or social implications of these systems. If we believe that the technical definitions of these systems is irrelevant when thinking about their ethical and social implications, then the limitations of technical fixes ought to be of primary focus. And I think that's kind of what I'll talk a little bit about, which is a few instances in which some of our work is helping us think about the limitations of these technical fixes or these statistical adjustments that focus on quote unquote bias. So one area of this work is with emerging work with my supervisor and colleague, Dr. Elizabeth Kazunis, who's a postdoctoral fellow at AI Now. And what we're looking at is trying to analyze the landscape of research practices that look at trying to understand bias and try to fix bias when looking at AI models or algorithms built on electronic health records. And what we see is that there is a lack of ingenuity around trying to address model disparities when those healthcare records themselves have gaps in patient records, primarily because lack of access to care, lack of trust. Some systems that were specifically built on trying to understand behavioral health outcomes on electronic healthcare data would acknowledge the limitations due to the fact that there might be omissions in that care record, primarily because some populations, some people would be concerned about their use and wouldn't report them due to stigma. This I also focus on underreporting of chronic pain. Cultural factors play an important role in how people interact with care systems. This is also building on work by Dr. Kazunis in other spaces as well, primarily originating out of the human-computer interaction space. And that's one area in which, as an institute, we've been focusing on interdisciplinary work in that regard. Another area where technical fixes for systems might be limited, I think emergingly so, is privacy. Right now, data privacy is a huge issue when it comes to building, especially healthcare algorithms and models. And some technical fixes have arisen to try to combat that, and differential privacy and federated learning are some aspects of that, in which we can mask or encrypt individual data points, but preserve underlying sort of data summaries for the larger data set, so that analytics is still useful, especially for population health. But I think what we're fast realizing is that there's a myth around de-identifiability. I think on the right, what you're seeing is a New York Times headline recognizing that MRI images of the face can be re-engineered to identify somebody. And that is data that doesn't have any personal identifiable information. And so privacy is another area in which governance models would need to focus less on the technical definitions of privacy and focus more on sort of data governance. Another aspect in which we're seeing the technical limitations of interrogating models is when thinking about flawed sciences and flawed uses. I think here particularly there might be more saliency with genomics and research around genomics, specifically around genetic testing and what that might look like, at least from a science communication standpoint, but also recognizing that data sets or data repositories of human genome sequences, particularly that, for example, of 23andMe, commercial entities, are subject to surveillance uses, especially with the police getting access to that data. And that will affect trust later on, especially for communities of color that are already over-policed and over-surveilled. And so that is another issue in which building these systems has limitations in which technical physicists just can't fix that. Here's a more egregious example of that, actually, where a company by the name of HireVue, which helps employers interview employees and find hirees online and provide software for interviewing over video conferencing, decided to create an AI system that would then help the employer identify certain metrics in the video conferencing or interviewers that are providing answers by doing facial recognition and then detecting emotion to see if those metrics of facial expression will then translate better to job performance if they're hired. And this was obviously, this is a much more egregious version of this, where the AI system is built on very faulty science and faulty correlations. And this specifically on the right is part of an excerpt from a complaint from the ACLU and the EFF to the FTC to try to crack down on this. But this also has profound regulatory implications for agencies like the FDA, in which you know, some of these systems purportedly can get released, and the FDA has no real way of interrogating them outside of the technical fixes, which simply don't reach the, I guess, threshold in which you would want to evaluate the systems holistically. The top is a excerpt from Aaron Brodwin's reporting in STAT on the number of different algorithms that hospital systems have been deploying to try to triage patients during times of COVID. And now, of course, some of these algorithmic systems or these softwares have been fast-tracked because we're in a pandemic and FDA has released certain guidelines around their clearing. But the bottom is another quote from Andrew Beam, which I thought was fascinating. And he says, I think there's a dirty little secret, which is if you're using a local model for decision support, you don't have to go through any regulatory clearance or peer review research at all, due to the fact that hospitals have their own data and can build these systems in-house. And so releasing them without sort of the regulatory oversight that you would normally want to have is incentivized in some ways. And so how do technical fixes address some of these concerns when they do have profound implications on bias is important. Now, I'm not talking at all here about intellectual property as well, which is another case in which how do you build these systems that then have access is another issue going forward, but I'll leave that for another time. And so what I want to do now is recognize that I've outlined a couple of different places that technical limitations have, technical fixes have caveats for them, and the need for interdisciplinary work to try to understand the larger holistic space of these ethical implications. What I do want to talk about is a little bit about how we're thinking about solving them or addressing those biases early on in the development phase. And this is coming out of some of the work that I'm working on, looking at AI and global health, specifically with my collaborators and colleagues of Tarshi Prokyasta and Judy Gichoya at Indiana and Emory. I'm trying to understand how have some electronic or technical systems been built in low resource settings that have addressed bias and sort of these larger social implications for their work. And so one of the things that we've come across has been two things. One is that AI systems that are sort of built and developed to try to replace expertise or try to fit areas in which capacity just does not exist has less saliency and less uptake compared to systems that are much more fixed on capacity building. And that agenda is defined or has been defined largely by the communities that require these systems in the first place. And so recognizing that what has been termed in the field participatory design, i.e. that communities that are impacted or seeing these systems deployed in their own settings, having a say in how they're developed is an important part of that. And so what we're recommending from looking at some of these successful implementations of mHealth tools is that participatory design has an interesting approach and a value in designing AI and ML systems for healthcare. Now, this isn't without fault. This is a paper by Kensing and his team in Norway that I think is really fascinating if you have a chance to go through it, detailing the story of building a commercially viable mHealth tool in the healthcare space by bringing those stakeholders, including patients, to the development table early on to try to identify where those bias aspects or those social implications might be able to be headed off before they're developed, deployed, and then realize that they have to pull back some of the problematic inferences that are being made. And one of the things that they notice is that in a multi-stakeholder setting, such as having patients, commercial partners, and physicians at the table at the same time, is that their needs are going to conflict at some point. For example, in this paper specifically, they detail how the ML tool is specifically about reporting for cardiovascular events for the patient, and so communication offline or online between patients and physicians. Patients really love notifying their physicians about what's going on, every little detail, and physicians found that becoming very problematic because there was a lot of noise in the data and it was just burdensome. But Medtronic, who was the commercial partner in that, really liked that patient feature because for them, the commercial value was the data that the patients were presenting. And that allowed for them to build machine learning models for other products. They were able to resolve this, but this was an interesting area in which commercial interests can skew bias and skew other ethical implications or workflow implications that may not be ideal. And this is something that I think, especially in healthcare, is very thorny and problematic, and participatory design at least helps bring to light. Here's another example in which that has had less of an issue. It had been much more profoundly impacted in a positive way. This is a report created by Madeline Eilish and Elizabeth Watkins at Data Society, working with the Duke Medical Center's team, especially led by Mark Sendak, on looking at implementing a AI algorithmic system to intervene for patients with sepsis, determining who would be at risk before terrible outcomes for the patient, and predicting how we can allocate that resource. And one of the things they found was that when developing those systems, recognizing that even though these systems are meant to be disruptive and change workflows in new ways, part of the implementation process, especially stemming from a participatory design approach, is wrestling with how to repair that larger political or larger upheaval of workflows. Specifically in this case, recognizing that nurses would have more power with physicians, and that created problems in communication. But how do you repair those forms of workflow? And I think that is where the really interesting work of dealing with the ethical implications comes to light, that doesn't see itself as sort of just fixing the technicals or fixing for statistical parity in the data set. And I think this was a really profound example of that. Now, to be clear, there were no commercial partners at the development of this system in the first place. So that might also play a large part in it. Obviously, I don't want to present participatory design as one fits all approach. There are issues in which participatory washing could be a significant threat, in which including communities that are being impacted, or including people that might be disproportionately harmed at the beginning, presents a way in which those concerns could be sort of washed or just used to be paid through lip service. If those concerns don't have any real impact in the development pipeline at the very end of it. And so participatory design has been a new and fast approach in the ML and AI ethics space. But there are concerns, specifically due to power and the politics that they play. And so I just want to end on that note, at least for now, really recognizing, at least for me, the work that's guiding how I'm thinking about these systems being built and developed is that you can't really excise the political implications and the social implications that are thorny when building these tools that don't really have a technical sort of vantage point, but that do require interdisciplinary work. And that's why I'm working at the AI Institute right now. I'm looking forward to being able to speak more with this with you all. Thank you so much for your time. I appreciate it.
Thank you very much, Varun, for an excellent talk. So I'm going to start with a question that maybe springboards off a question that Subhashini Chandras Kiran posed, I think, for the last speaker, and we'll come back to Q&A, but really a little bit more tailored for your talk, which is you noted that we should be wary of participant washing, as you said, in community engagement efforts. So do you have thoughts or examples of what more effective community engagement strategies might look like, or at different parts of the development pipeline?
Yeah, absolutely. Thanks for that question. I think if I'm thinking about it, and to be quite honest, this is partly in the early research phase, so I don't want to be too prescriptive. But from what I've seen in terms of successful implementations, and I'm specifically thinking about the sepsis watch algorithm at Duke Hospital System led by Marcus Sendak, as well as two of the examples that we highlight in our workshop paper on looking on global health implications. One was having clear guidelines around what the commercial incentives and who's coming to the table is really how you gauge that, and who defines the agenda, right? If the agenda is about building a tool that is meant to optimize resources on the health system's point of view, right, then whoever you bring to the table is not necessarily going to have the best leverage in defining that agenda if it's heavily skewed towards hospital use. So that's where participatory washing can become a threat. Whereas if agenda setting comes from the ground up, whereas patients are able to define that need just as much as other players at the table are able to do so, then I think there's a chance for bias and transparency and fairness to be wrestled with upfront. So that's those are the kinds of things that I'm seeing work from the beginning. But there might be other ways, and I think that's ongoing work, and I'm excited to see what comes out of that.
Great. Another question that came up in the box, so it wasn't so much a question, it was a comment from Heidi Sophia, and the question of privacy, since there is no true anonymization, just de-identification, but we're coming across myriad ways now, and this is certainly something we've tackled in the genomics community for many decades, about re-identification using biological data. Now we can think about this using imaging data, using natural language processing over text data in health systems, and how these things can compound. So what are your thoughts on how we go about assessing, measuring, and what are the balances of risk versus benefit in that scenario?
Yeah, so this is a really interesting problem. I think for me, the way I'm thinking about it is more in the accountability and regulatory space. I am also someone who, looking at some of the research right now, believes sort of in like the myth of de-identifiability in this current world that we live in, where disparate data sets, I think it's already been shown, and I'm thinking about Zach Cohen's paper, I believe, that does this, where they took two different data sets, stripped away personally identifiable information, and they were still able to link records, right? And that's just, one clear, that's a very isolated example. I think linking data sets and linking information from a variety of different places is already happening at the federal level for a variety of other things. And so protecting patients' privacy using federated or distributed learning systems can only get you so far, I think, based off what we're seeing. The best way I would say about thinking about that has been trying to step away from individual privacy, because I think what ends up happening is that once you only frame the problem as an individual patient record privacy problem, you are inherently going to start increasing monopolies around that, right? So whoever actually owns the data then doesn't have to share that, or whatever can be. And so therefore systems, and this is where big tech, I think, is going to really feast, in which they don't need to worry about that at a particular level, especially when they're building partnerships with health systems, and they just want to acquire the algorithmic model rather than the data set themselves, which they can do. And I'm thinking primarily about the issue with Google and partnering with Ascension as a health system, where individual health privacy was addressed, but the larger issue of antitrust wasn't. And so who gets these models and who gets to build them, and that power dynamic then gets shifted to the wayside if individual privacy is necessarily put at the fore. And I think doing that also allows you to protect patients adversely affected by models.
Thank you. So we're going to have to wrap it up now for time, but we'll come back to some of those questions in the questions later. Okay, thank you. Back to you, Dave.
Thanks so much, Varun. Our last speaker of the panel is Danton Char. He's an assistant professor of pediatric anesthesiology, perioperative and pain medicine at the Stanford University Medical Center. And in his spare time, he thinks about machine learning and AI. And so in the clinic, and so take it away.
Thank you very much for having me. I'm going to talk about identifying and anticipating ethical challenges with machine learning for genomics, ideally before they become consequential. Much of what we know about artificial intelligence, or at least what we're thinking about within the healthcare space, comes from non-healthcare contexts. So 2017 was a very bad year for AI and ethics. We already knew, like this example from the often cited ProPublica article about the bias of the Compass recidivism software, that bias inherent in training data could be reflected in an algorithm. Very quickly in 2017, we also had the Cambridge Analytica Facebook scandal that raised issues about privacy and targeted information, as well as the Uber's gray wall algorithm designed to avoid regulation, and the Volkswagen emission scandal, in which they built an algorithm to try and avoid emissions regulations as well. It didn't take much reading into the Mueller report to realize that there were also very significant geopolitical algorithms getting deployed to try and target information sources within the United States, as well as potentially infrastructure within the United States. This all holds true within the genomic space as well. This is the last month's report from the Director of National Intelligence, looking at collection of genomic information by other actors around the world. I don't mean to signal out China in particular, and I don't think it's alarmist, though, to say we simply don't yet know the full extent of the risks to individuals or to groups of individuals posed by either multinational corporations that are collecting precision health information and genomic information and attempting to monetize it, or by other nation states that don't reflexively adhere to Athenian ethics filtered by the British and American centuries. Even if you don't think that geopolitics or economics are really the direct focus of the ELSI area of research, I think that the trust questions raised by this type of ongoing collection of data are. I want to take a brief detour into my own research. So my K award from NHGRI has involved looking at implementation of genomic information into the care of critically ill children, particularly children with congenital cardiac disease. One of the surprising findings from our research is that for bedside clinicians, the chief use of or appeal of use of genomic information is allowing earlier declaration of futility, as well as informing decisions about withdrawal of care or rationing of scarce resources like organs for transplantation. And this is a neonatologist saying, for us, one of the more immediate benefits of genomic sequencing is we could have that discussion with the parents and change our goals of care to comfort as opposed to prolongation with futile intensive care. These findings have borne out as in implementation studies of as genomic sequencing has been deployed in pediatric critical care contexts. Clinicians have found the use of genomic information for withdrawal of care to be the majority, the most useful use of that information. We've now completed a national survey looking at how clinicians caring for children with congenital cardiac disease would change their clinical recommendations based on genomic information they receive. So this is for a child with hypoplastic left heart syndrome, clinicians' likelihood of recommending surgical palliation, ECMO, or transplant candidacy based on genomic information that reveals either a chance of breast cancer, a childhood onset cancer syndrome, intellectual disability, or autism. This is also from that survey looking at genomic information that would reveal severe DiGeorge associated schizophrenia and the impact on the decision to recommend either ECMO or transplantation. I'm not trying to vilify clinicians. All of these decisions are being made in the critical care context where there are scarce resources and tight benefit to burden ratios in many of these therapeutic decisions. And I think what we're dealing with are concerns that are ethically unresolved, which is to say it may make perfect sense not to offer an organ for transplantation to a child with a childhood onset cancer syndrome because we know that the immunosuppressive medications accelerate neoplastic disease. However, not offering ECMO or not offering surgical palliation are ethically more problematic, similar to not offering transplantation or ECMO or palliation to children with severe intellectual disability. And it requires some discussion because it's ethically unresolved. Having these ethically unresolved concerns in this space has led to some issues of concerns about power disparity and concerns about distrust in parents. And these are interviews from studies we've done with parents. This is one mother saying they've probably spent over $4 million on her child just in the 2 years of the transplant, pre-transplant, post everything. If we had known about his mutation, his genes being bad, if we'd known about it in advance, I always think, what if they had declined to treat him? And another parent saying, if there's pre-existing conditions or the potential for conditions to come up in the future, how much does a medical institution invest in helping somebody that's potentially going to die? This is another parent stating explicitly, we see distrust across the board in all of our institutions. You see it with the measles outbreak, the anti-vaxxers, there's distrust in pharmaceutical companies, there's distrust in the mega industry of healthcare. This will all get worse and more intense with genome testing. In many ways, I think that the critical care space is a microcosm of healthcare more broadly. It deals with issues of difficult ethical decisions, resource constraint that we have now unfortunately seen over the course of the past year with the pandemic. We've seen our entire healthcare system have to face resource constraint and difficult decisions about allocations of resources. And I think what we're seeing is increasingly august bodies like the American Medical Association now acknowledging and expressing concern about the erosion of trust in healthcare, as well as what strategies need to be put forward to try and combat the misinformation that feeds upon this distrust. So now that we have a sense of what we don't want ethical concerns with machine learning to look like, what do we want machine learning to look like in an ethically appropriate way? This is Alka Patel's work from the Department of Defense. In 2018, they recognized that there were concerns about use of artificial intelligence in modern warfare and governance, and came up with these 5, through modified Delphi studies that they did at several institutions around the country, came up with these 5 principles that they thought ethical artificial intelligence should adhere to, that it be responsible, that humans should be responsible for the outcomes, that it be equitable, that it avoid harm and unintended bias, that it should be traceable, which is to say that it be transparent and auditable in its methodologies, its data source, and its workings, that it be reliable, that we know where it should be deployed, and that it be safety tested across the entire cycle, of use in that domain, and that it be governable, that you could detect unintended harm, that you could detect when it was not doing what it was supposed to be doing, and that humans could disengage it. In 2018, also, Michael Abramoff got approval from FDA for the first fully autonomous diagnostic system in healthcare. This is the IDX system to diagnose diabetic retinopathy. As part of his approval process, much of the scrutiny from FDA focused on ethical concerns. And working with Michael, we came up with these 3 additional principles we thought were appropriate for machine learning tools being applied in the healthcare space. The non-maleficence, that it not do harm, that patients benefit, that it improve clinical outcomes, that autonomy, that patients still controlled their healthcare, that liability for the AI system malfunction should be related to the degree of autonomy with which it's used, and questions of ownership of data. Who owns the training data it was based on? Who owns the data that it collects and generates? As well as equity, absence of bias, fairness in distribution, and access to benefits for all groups. Earlier this year, with the National Defense Authorization Act, White House interagency coordination of AI, including over ethical concerns, was created. I don't know if the principles from the Defense Innovation Board or the FDA will be the end point of what we think ethical AI will be, but I think it's likely that the White House interagency coordination will draw on the FDA and Defense Innovation Board work as what we think AI should do moving forward. This is a quote. There's an old saying that a problem well phrased is half solved. This much is obvious. What's not so obvious, however, is how to put a problem well. I got this from Chris Feutner at University of Pennsylvania. And I think in many ways, the easier part of the problem may have been figuring out what we want AI to look like. What's harder is that we don't have a methodology to how to identify ethical problems emerging with AI being deployed early. The current model for ethical evaluation is largely a cadre of expertise or of experts who evaluate ethical problems. And there are several concerns with this, but chiefly for the deployment of machine learning, which is happening on a broad and vast scale, it simply won't scale to the volume of AI that's being developed. In the same way that we can't have just 1 or 2 experts at any institution who know how to do a randomized controlled trial, I think we need to have, as part of implementation science, some component of implementation evaluation that evaluates ethical concerns early and gives space to affect change. Chris Feutner, Michael Abramoff, and I put forward this beginning of an idea of what such an evaluative methodology would look like, anchored in, and this is the left column, the idea that design and development of any new technology moves through a series of processes from conceiving the idea to ultimate implementation. And that as part of each of these stages, a series of design questions get asked. And that these design questions can have ethical implications. And that how different stakeholders affected or designing the machine learning tool or using the machine learning tool or deploying the machine learning tool would answer these questions reflects their different values. So to use this idea does require 6 premises, which I believe, and I hope that you will agree with, which are that multiple stakeholders can be impacted by any machine learning tool. And these stakeholders can be identified by examining the design and deployment context. Stakeholder groups have different values and explicit or implicit goals for a machine learning tool that should and can be ascertained. The process of design and development of a machine learning tool involves making a series of decisions. How a stakeholder makes these decisions or would want these decisions to be made reflects their underlying values. And this draws on work in values and design by like Shilton, Bray, Manders-Weitz, Johnson, that where stakeholder groups disagree or their values are at odds about resolving these decisions, where values collide are where ethical problems are most likely to emerge in future. And that while some of these value collisions may mark novel ethical concerns that require a cadre of experts to address, many can be resolved by drawing on prior scholarship on similar or related problems. And that simply identifying them is a long way towards addressing them. So this is an example from non-healthcare. This is the first self-driving car accident with an Uber self-driving car where a pedestrian was hit. And in analysis, it was felt that Uber drivers had wanted to smooth the ride and had to do so had reduced the number of false positives. But obviously in reducing the number of false positives, they corrected to a point below which it failed to recognize a pedestrian at night walking a bicycle. And certainly, although the stakeholders involved with the design of the car and deployment of the car wanted the smoother ride, other stakeholder groups that are potentially impacted, pedestrians, would have wanted a higher safety threshold. And so this is the kind of value collisions that then could have ethical implications. As I've done additional work on mortality prediction or just general predictive analytics, this is with Nigam Shah, I think we've reduced it to 3 interacting data elements in the predictive analytics space, which is where ethical concerns can arise, which are the model and the output it provides, the workflow into which the model output is introduced, and the policies for allocating an intervention at a certain output recommendation by the machine learning tool, and the benefit-harm trade-offs of the recommended intervention itself, and that value collisions can occur at any of these 3 elements. So I want to briefly touch on a case study as we've tried to analyze deployment of a machine learning tool using an anticipatory methodology to try and identify addressable ethical concerns early on. So this is a mortality prediction algorithm designed to be used. So when patients are admitted to an inpatient medicine service, the algorithm, they tried several different models to make this algorithm, but the overall goal is to identify the likelihood of 3 to 12-month mortality in the being admitted patient as a marker that the patient could benefit from a conversation about advanced care planning. And it stems from a very laudable goal, which is that most Americans would like to die at home, but many Americans are still winding up to die in intensive care units because of sort of poor recognition of mortality and poor advanced care planning. And so this is an attempt to try and address that concern. This is work by Natalie Deutsch, who's working with me. She's now here at NHGRI, looking at all of the stakeholders impacted or potentially impacted by deployment of this machine learning algorithm, design and deployment of this machine learning algorithm. And she kept joking that this is like one of those, in those detective TV shows where they have like the string attaching to the different photographs, that this was our detective web of everybody who was impacted. So having identified these stakeholders, we then conducted a series of interviews of clinicians, designers, administrators, as well as patients, and identified multiple areas where they had value collisions. And in the interest of time, I won't go through each of these details, but there were 3 big areas where they ran into collisions. One was perspective on the use of mortality prediction for end-of-life care. And the collision is, palliative care clinicians pride themselves on their prognostic skill and felt that having numeric support in the likelihood of near-term mortality would benefit them. Other clinicians felt quite strongly that whether a clinical gestalt is being used or data is being used, there's going to be a ceiling on the accuracy of mortality prediction, and that there's an inherent trend in numeric exceptionalism that people are more likely to believe a numeric output than they are necessarily to believe someone's clinical gestalt, and that clinical actions might be taken that could be limiting to someone's overall life by having this numeric mortality numeric in the chart. Related to this within the workflow was the question of who actually got the mortality prediction. Should it be delivered to patients? Should all of the clinicians involved in the patient's care have this mortality prediction? Should only palliative care clinicians get it? In the original implementation model, the mortality prediction was only going to the palliative care service, who were then using it as a screening tool to identify patients who might benefit from advanced care planning. And a concern about introducing a mortality prediction into the clinical space was that there could be unintended uses. And then another concern of the benefit-harm trade-off was just because of the speed with which machine learning tools are being deployed and the overall attention they're receiving, was how to protect the machine learning mortality prediction research from external pressures like administrative or social media scrutiny before the research was actually completed. And this is a quote from one of the designers explaining their interaction with administration. At one point, they were asking me if we could successfully predict if patients have less than 24 hours to live. Because if they've got 24 hours or less, we're going to admit them to an observation bed and not actually admit them. An observation means they're not officially admitted. And if they die in observation, they don't count as a death against regulatory measures. And I was like, I feel like I'm going to vomit in my mouth right now because you're telling me you want to know they're going to die in 24 hours because you wouldn't put them in a normal inpatient acute care bed. You'd put them in observation. So based on our analysis, the design team was able to prioritize needed efforts focused on 3 areas. One was examining alternate implementation strategies to delivering the mortality predictions, which they're going to into the workflow, which they're going to continue to do to see, well, should the bedside clinician get it? Should a counseled conversation with the patient be where the information gets delivered? The other was almost a branding concern, that if they were concerned about unintended uses of a mortality prediction, which the designers were saying, well, it's only validated to be used for advanced care planning, maybe they should stop calling it a mortality prediction and should just call it something like an advanced care planning needs probability, and eliminate the confusion that was arising in clinicians and patients' minds about, and administrators, that it could be used as a general mortality prediction. And another that we will all need to consider as we move forward is how to shield ongoing research into mortality prediction from social media or premature scrutiny until endpoint-driven studies are completed, in the same way that we have protections similar to blinded clinical trials. Obviously, concerns that we need to continue to work on are, when should future ethical analyses be conducted? Should it be done iteratively? How do we make this part of implementation science analyses of machine learning deployments? How do we study algorithms across entire learning healthcare systems as opposed to simply just individual deployments? And how to better streamline the ethical analysis process. Obviously, there's very labor intensive identifying everybody and doing individual interviews. Can we consolidate the questions? Can we deliver them? Which questions are highest yield? Can we deliver them in other ways? Can we figure out optimal number of stakeholder assessments needed? And these are my acknowledgements, my obvious gratitude for the K Award from NHGRI and the Human Centered Artificial Intelligence Seed Grant, as well as everyone who's been involved in all of this research. Thank you very much for including me.
And thank you so much. We've got a couple of questions in the chat that I kind of want to hold for the broad discussion. So I'm gonna ask you a couple of things. Maybe these aren't easy ones, but do you feel? I guess I'm going to turn one of Courtney's statements into a question. Do you think that with the complexity of the clinical genome, physicians can benefit from ML or AI assisted tools in clinical genomics as a clinician who's been looking at these applications?
Oh yeah, I mean, absolutely, right? The downside of being, right, the joke is if I was an optimist, I wouldn't have become an ethicist. But absolutely, I think the promise of machine learning is that it will allow, algorithmic tools in general, is it'll allow us to process this big data that constitutes genomic information that's part of precision medicine, to allow us to make connections amongst the data that no individual clinician, or even from the public health tools we've had over the 20th century and into the early 21st century, have been able to make. So yes, absolutely. I'm very hopeful for what machine learning and AI can do.
Great. Another question, kind of the same question came up twice. In your evaluation implementation science approach for anticipating ethical issues, where there are these value collisions, how does one address inherent asymmetries in power?
So a great question, and I don't. I would rather it be thought that what we're trying to do is a starting point, not an answer. I really want the take-home message from the talk to be rather that we need a methodology and it needs to be something that could be more deployed than that we have fully solved it. I do believe one of the problems that needs to be discussed with AI implementation everywhere and machine learning implementation everywhere is exactly that. I know that Dr. Sankar spoke about clinical deployments of AI and machine learning tools, but I think that inherent in this power disparity is this issue of that many of the most clinically impactful deployments are in fact going to be things like allocations of material supplies and financial costs that occur in the background of healthcare delivery and yet are fundamental to it and are very easy low-hanging fruit for machine learning tools and are already being deployed across many industries in that way. And those power disparities, I think, are going to have to involve an open identification, like just Varun talked about before, that we'll have to identify who all of the stakeholders are and what their likely beliefs and what they want out of the deployed system to be.
Thank you. That's a very thoughtful answer. I guess maybe just touching on the example you were touching on at the end. Well, let me ask a different question. You know, you and Varun both sort of, actually everyone sort of talks about the importance of the context, the clinical context that these algorithms enter into, and you know, and you can certainly break the context or the finding, you know, that the machine learning comes up with just may not fit into the healthcare system. And so, you know, our healthcare systems are incredibly complex, right? And heterogeneous. Do you think that machine learning's ability to incorporate and address heterogeneity is something that can help to overcome, you know, can the conclusions that a clinical decision support algorithm come up with, can they incorporate and transcend the heterogeneity of care, or is the and is that a strength of these applications, or is it too much to overcome?
I think unfortunately my answer is yes and yes.
Yeah.
I think that we're in a tremendous sea change now. You know, much of the ethics that we've thought about in healthcare has been largely dyadic, right? A relationship between a patient and a clinician. And I think that we're now in a sea change where really the relationship is between a patient or a cohort of patients and a learning healthcare system model. And I think that these machine learning tools, algorithmic tools, are fundamental to the successful implementation of a learning healthcare system, which has all of the promised benefits that we were talking about for genomics, of being able to analyze data from 30,000 feet in a way that no individual clinician could ever see, and yet also will have real problems in the delivery for the individual patient and making sure that all of the protections and the compassions are inherent. I want to touch on that too, because I think it's fundamental to the problem that you guys are having, like with the All of Us project as well, which is, you know, the very tension is, how do you get successful access to all of this data and use all of this data in the way that it promises to be used, while maintaining the protections that you would want as an individual, without making those protections so restrictive that the research or the promise of analyzing the data can't be fulfilled.
I really appreciate that response. With that, I think it's, if I'm not mistaken, I think it's about time to move to general discussion. So maybe we can bring Varun and Pamela back and dig in.
Great. So I'd like to invite Danton and Varun and Pamela. I cannot see you, Pamela. I know your video may not be working. Okay, wonderful. Back. Well, Pamela, just to let you know, you are there. Great, perfect. Okay, so look, just a fantastic job by all the speakers and some really lively discussion, and it's about to get livelier. So, you know, much of the three talks that we heard this session were really focused on healthcare applications, where we all know that a lot of the time the stakes of getting the wrong answer are pretty high. Yeah. So just to kick this off, thinking about where we are in the field of ML, AI, and precision medicine and genomic medicine, who do you think are going to be the early users in the health system? And is it likely to be providers, or is it likely to be other users? And, you know, maybe even to flip that question on the head a little bit, thinking about where are the thresholds where getting the wrong answer might be less of a concern, or where are the thresholds where getting the wrong answer is of the highest concern? Who would like to jump in first? Varun?
Yeah, I can jump in a little bit here, just based off of what I'm seeing in terms of that. I think the adoption by providers, specifically within, you know, the public-private research partnership, is going to probably get a better long-reaching start than I think some startups that are trying to capitalize on this early are getting. I'm, you know, thinking about a couple of startups that are trying to do, you know, machine learning around predicting, you know, certain genes for a couple that's expecting a baby, for example, and that being very precarious, right? Because the science behind that's a little faulty. And I don't think that they're going to get very far, A, from a regulatory standpoint, but also B, from a trust issue. So I think where the NIH and other places see potential in funding and bringing that to laboratory science, I think that's where the potential is going to be. I think the first real use case of this is going to be.
I suppose one question is use in what way? Because as I said, I think that the clinical applications are going to be slow in coming. And I think that given the kind of work that's required to create them in a way that is useful, it's massive. And therefore you can expect that it's health systems that will eventually take something that looks like a good idea and turn it into actually the new routine way to handle admissions that look a certain way or discharges that look a certain way. And so it's health systems that have to decide to adopt it. And I think that there's certainly, and Danton made this clarification, which is useful, much what I talked about was clinical applications. There's a lot of machine learning integrated into healthcare already on the operations side, scheduling appointments, making sure that the, parking lot doesn't get too full, making sure supplies are available, all of that. So there's a lot of it there. That's not really what I've been talking about. And of course, it's important ways other than just those ideas. Payment, insurance, et cetera. But once you get to payment insurance, you have a pretty close overlap with clinical things, clinical issues. But I do believe that it's going to be sporadic and it's going to take a while in the clinical setting. But maybe others disagree with that.
Certainly, I agree that one of the biggest problems is going to be not just the algorithmic output, but its impact on workflows. Absolutely. And I think what's so challenging for us is in many ways, a lot of the ethical problems we're looking at aren't that are sort of indistinguishable from whether the algorithm actually simply does what it's supposed to do. And I think that distinction is going to continue to be a gray area for a little while, or does what it purports to do rather,
right.
not necessarily what it's supposed to do.
Yeah. So
and then what's very important here is that it's going to take a long time to be able to see that. And the issue vis-a-vis health disparities is what then becomes very important, because it takes a long time for these things to play out, which is why I think it's really important that people put thinking now into formulating not better necessarily, any very clear strategies about what it means for ML to take not just health disparities, but to take the goal of promoting equity, to use that as a motivating goal that is apparent across the pipeline of the development of these technologies. Because to wait until you actually see how it plays out, which in some sense we have to no matter what, but to wait to see what happens is going to be a mistake. And I think that that's an important feature of all of this.
Yeah, certainly in our work, looking at you talked about reification. I think, looking at mortality prediction, that's a real concern, because when people have started trying to use mortality prediction for, say, allocation or decisions about what therapies to offer for, say, oncology or radiation oncology, if you use an all-cause mortality prediction, that has baked into it health, wealth, race disparities, and it doesn't make the algorithm output wrong, but it also means that someone who has those disparities disadvantaging their mortality simply doesn't get the more aggressive treatment. And how we address that inequality up front is going to be a real issue with these tools.
Yeah, I mean, that's certainly something, Pamela, that you said, and I'm sort of paraphrasing, it's something along the lines that efforts to improve population health have to go beyond minimizing harms and actually have as a goal achieving health equity. I hope I didn't butcher that. But also, Danton, in your talk, you emphasized this as well, that we're really, that's a goal, but we're really lacking the methodology for metrics of success. So we don't know when we've achieved it. So what do you think would be the maybe good enough measures that we could start to deploy, or is there any, so that we can try and get this trade-off for, we may not understand very well, but we have a goal of detecting health disparity early to course correct early. What are some approaches that we might take for that?
Well, I think the most important thing is for the machine learning, the people who are experts in machine learning now, for them to think through what are the metrics, what are the features, how are they going to evaluate it as they are creating the machine learning. And yes, you're right, we absolutely should have metrics to evaluate these technologies once they are deployed into the clinical setting or wherever they're being used. But I think my point now is that the onus, I don't know the onus, but where we need to be looking is to encourage, facilitate the attention by people doing the machine learning now to figure out exactly where these things need to be integrated. Because to create a machine learning algorithm and application it's multiple steps. Every step is a decision. Every decision has a value embedded in it. And somehow there needs to be a better theorization to guide machine learning workers so that as they're making those decisions, they have a sensibility directed there so that they can think about those things as they are creating them. And then, of course, you have to test it. You have to, implement it and see whether what they thought would work does work.
Yeah, actually, that brings me to a question from Laurent Philippe Albo, and I'm just going to paraphrase this a little bit, but really, it's about tying the whole part that the AI ML community needs to learn about ELSI and the methodology there, but also the other way around, how the ELSI community could learn about aspects of AI and ML. And actually, he's making a point that, that actually the community think about ML and AI very differently and maybe even in nuanced ways. How can that community provide a better explanation of those tools and better ways, methodologies to assess them, etc. So how do we bring, I mean, there are efforts like this, of course, but how do we continue to foster bringing together communities, not just exclusively to ELSI communities and AI communities, but all the other stakeholders?
So I can answer that question as someone who's done that. As Danton pointed out, yeah, I'm not a doctor. I'm a software engineer by training. No, I appreciate that. My dad's probably howling watching this live stream right now. But I think for me, the way I've sort of maneuvered my machine learning expertise into trying to integrate how my skills is in trying to build or think about applications has been really trying to foster larger discussions around interdisciplinarity, but also building those diverse development teams. And so just building off of like understanding, what are those metrics? Because I think that's kind of inherently tied to this question. Recognizing that building a model that can beat a benchmark or a couple of physicians or is represented by an AUC score of whatever is not going to necessarily translate into the clinical setting. And that is actually some fact that has to still be understood by the computational field, right? And that's not just a fact of learning, that's a cultural difference. And those cultural differences in disciplines need to be bridged in a way. I also think at some level, there does need to be some sort of federal regulatory guidance on this, because there's a lot of silos that are being built around these, and different hospital systems have their own way of dealing with this, which I think is great. But discussions like these that lead to some level of federal guidance on this, because I think the FDA is also kind of step up on this level as well.
Great. Okay, so I want to hop a little bit to the Q&A board, and one of the questions that got a lot of upvotes was from Mildred Cho, and it really is dealing with conflicts of interest. How can any conflicts of interest between developers of AI and machine learning and the end users in healthcare, how could these conflicts be mitigated, particularly as we're in the absence of robust regulation and evaluation and governance? Anyone care to?
Yeah, I can jump in if just as a first case. I think one of the interesting things that we've seen has been the way, so I think this is pertinent when it comes to big technology companies partnering with hospital systems. There seems to be confusion around what kind of data is being shared. I'm thinking about the NHS and Google's partnership. There being confusion around patient data being brought into Google after Google acquired DeepMind. So the acquiring of companies and how does that data, patient data, travel in those instances is not clear. That information is also not clear in the business contracts that many of these companies signed to. So there was a whistleblower actually that reported in The Guardian on the fact that Google developers could see patient data when working with Ascension, right? And so I think that's kind of the conflicts then arise with, well, what are the actual incentives that are building these partnerships together, and who's getting what out of what? And I think potentially borrowing conflict of interest practices around how doctors maybe report around their funding from pharma, et cetera, there may be something there. As someone who's just kind of been trying to understand from what's publicly reported and how to tie those things together, that would be, I think, a start.
Thanks, Varun. Any other thoughts? Go ahead, Pamela.
I think this is a very rich and complex area of what's important to look at in the ethics surrounding machine learning in healthcare. Certainly one strategy is to just make the whole concept of conflicts of interest more obvious, more known, more knowable, more accessible to people working in machine learning who have not come out of healthcare. Mm-hmm. So the whole concept of you really are supposed to serve one master, not two, where does that come from? I mean, it comes out of the relationship between the physician and the patient. And although that is mimicked elsewhere in our society, that is one of its most crystallized places, law also. But I'm not sure that within machine learning as a part of training, I don't know that the concept of conflict of interest is very well is discussed. I'm going to guess that it probably isn't. So part of what you'd want to do is to work on training programs or curriculum or articles, joint articles, things like that, to just bring the awareness of the concept of machine learning. I also think that the idea that most or much of this work is done under the auspices of QI and QA raises many, many issues, because to the extent that the QI and QA is overseen by the healthcare institution, and to the extent that the healthcare institution is trying to cut costs, which is in itself not a bad thing, but one can imagine that there's your conflict of interest right there. You're supposed to be caring for the patient, but you also want to save money. Not that they are by definition in conflict, but you can imagine that they could easily become in conflict. And so I think that a lot of what has to happen is really, to begin with, a very clear description of the novelty of these intersections that happen now in machine learning and healthcare. This is really a different landscape. There's now this other entity, it comes in, it's fully formed largely from the outside. And as we've been talking about, there's questions of interpretability and all, but the point is, I'm trying to say is that it really is a different landscape, and it is something that needs description and articulation. And I think that is the first step toward trying to respond to what are potential problems.
Yeah, thanks, Pamela.
Also teach big tech what conflict of interest is.
Well, that actually brings me to another question, and the concept of profits and costs has actually come up a couple of times. But another question, because big tech is a big player in this, has come up throughout the three talks. Does any of you have direct or indirect experience with working in an environment, or an indirect experience of big tech who has entered into an agreement with a health system or an alliance with a health system? And what did that look like? Maybe the answer is no. Not yet.
No, not yet. So I was associated with Microsoft as their data science for social good fellow, but we didn't work with hospital systems on developing machine learning tools for public policy planning. Though I should probably talk with the team members that did.
I want to throw it a little bit back to the board because we've just got some great questions coming in, and one of them I just saw from Elizabeth Green, and now I can't find. Oh yes. Okay, so how can physicians and others find incentives to legislate or set policy? For example, that all machine learning and artificial intelligence must use open source coding and transparent algorithms and all those types of good practices, fair reproducibility, and or that corporations are prohibited from developing medical applications because they lack a required healthy conscience, for example.
We certainly are probably going to wind up, you know, I mean, the AI machine learning tools are not distinct from other technologies. We certainly have precedent for other technologies that are regulated differently in different nations or different economic communities, and that's probably likely going to happen with machine learning tools as well. I think this touches a little bit on Dr. Cho's comment and, you know, what both Varun and Pamela were saying and were asking, which is the cost and the financial components and what they impact. I mean, obviously the chief driver of these tools moving into healthcare space now is either profits can be made through synthesizing data in a way, you know, providing a service that previously has been unprovidable, or by streamlining and downsizing costs by reducing previously very expensive labor workforce. And so when harm can be demonstrated by either of those, whether the harm is economic, it will probably bring the companies into line to address those concerns, or if the harm is really direct to patients and unconscionable, will probably bring up regulatory control of these. But until either of those, I think that those are the main paths that we'll have to try and address concerns.
How does that play out in a landscape where early either big successes or big failures are likely to be more heavily weighted and will have a bigger impact either on adoption or non-adoption of some of these solutions?
Well, both. It's probably going to lead to a context of timidity and temerity, which is to say, I think that may be part of the driver as to why there isn't as much aggressive clinical pursuit into the clinical space. Because simply it's not as low-hanging fruit. And I also think it leads to a variety of problems, right? So like the IDX system that I was part of, the goal is to try and capture patients who previously are not accessing healthcare for diabetic retinopathy screening, right? The goal is to increase the catchment of marginalized populations. And yet, by definition, once you bring them in, because they're marginalized health populations, simply diagnosing them is inadequate. But now you've taken ownership of the problem, right? So if you go to a third world space, or even if you go to many slices of America, although you can say, because this thing is at whatever the local pharmacy is, that they have a diabetic retinopathy, that doesn't mean that you can materialize a workflow pipeline of ophthalmologists to actually treat them and provide care. It's similar to the problem with many of medical missions diagnosing congenital heart disease in third world nations. It's simply because you can diagnose the disease doesn't mean that you can get the infant or the child to some sort of healthcare system where they could provide a necessary intervention or necessary surgery. And I think that if people are forced to take that ownership or ethically, because they probably should take ownership for that, I think many people will stay away from aggressive pursuits to increase that catchment of otherwise marginalized groups.
So just to follow on to that, you know, we heard a lot about lack of access, underreporting, underrepresentation, certainly in genomics databases for big swaths of the population for many complex reasons. So what might be some of the non-obvious ways that that set of circumstances could compound in the utility of MI and AI in healthcare? Do we think that we can anticipate what's going to happen or not? Go ahead, Pamela.
If I understood your question, one of the ways that this can get compounded is because of the rhetoric that has surrounded machine learning, which is very similar to the rhetoric that surrounded precision medicine, that it's going to transform medicine. It's going to have better care and cheaper care, and this is going to be the next thing that will save the healthcare system that has so many problems. That has also been said very specifically in the context of precision medicine, specifically about health disparities. Join this effort, all of us, the effort, or similar things like that. Join this effort, donate your information and your DNA and your other biological samples and specimens, and we will improve, we will mitigate health disparities. Some people go so far as to say we will eliminate them, but let's just stay with the middle one. We'll mitigate them. Well, as machine learning rolls out as the next big thing, if this doesn't happen, if this is not made good somewhere, somehow that you can really point to, then what we will lose is trust. And you know, we're on a thin line there with whether or not we have public trust right now, but it certainly was built up. An enormous amount of effort was put into this for the precision medicine effort. And that has sort of slightly stepped back now that machine learning is what people are talking about. I mean, it is as active as a you know, kind of research and kind of healthcare. It hasn't stepped back, but in terms of public perception and public coverage, it's, you know, machine learning is what you read about now. Well, if we get through the end, if we get to a certain point in machine learning and not much has happened, then that undercuts all of the promises made in the name of precision medicine. And if we undercut those problems, we don't make good on those problems, I am very pessimistic as to the kinds of support we can anticipate from the public going forward for these efforts.
Great. So I'm gonna, oh, go ahead, Varun.
I was just going to say, I 100%, I think Pamela put that really well. I think in the long term, there's going to be that fallout of trust if those examples don't surface. I would say AI Now Institute in 2018, we created an algorithmic impact assessment framework for public agencies as well as private agencies who want to start thinking about how to be transparent around auditing so that there's more transparency around how they're procuring. I think that might be a part of that for sure. The other flip side, of course, is that just as we acknowledged, and I think Danton brought up really well, is that AI systems are being used all the time in the back end for organizational purposes. I think those might be happening and working really well. And so there's the adoption and the trust issue as well. It works well for you, but what about for us? And that might spur on a little further on if that's not the case. But I think Pamela put it really well, and I couldn't agree more.
Great. So I'm going to go back to the Q&A board, and maybe there's a lot of questions coming in about regulations. I'm going to maybe try my best to distill this into two parts. One is there examples of existing regulation and legislation, maybe some other domains that we can co-opt or tailor into this domain to expedite that? And the second part of that, is it possible to craft policy with enough teeth to get through all these built-in layers of obfuscation and heterogeneity in the systems? Who's brave enough?
Well, there is policy. There is policy now. I mean, the FDA is rolling out its policy, and I have to say, I know in the last 3 or 4 months they've made some changes, so I should be better up on it, but I'm not. But they've been testing different ways of regulation, and the very specific distinction is the way that the FDA has governed or managed regulated medical devices in the past was based very much on the assumption that the device was a physical entity, and that you assessed how it was researched and how it was tested. And then as that entity, it went out into the market. And of course, they might make small changes to it, but essentially the entity was the entity. And as it was dispersed across various clinic settings, you could see how it worked, and you could assess it and say, oh, well, you know, post-deployment, post-marketing, there's a problem, and bring it back in and assess it. The problem, of course, with machine learning is that by definition, not all, but most of it, and certainly one of its strengths, is that it continues to evolve and to iterate. And so what they decided to do, at least as a test, is to certify the company, certify the people, certify the process through which the machine learning is created. And so if it hits certain benchmarks, the idea is what they have created will be useful, ethical, safe, etc., effective. Yeah, I think that's a really complicated thing to do, because that's one of the things we've been researching. We've been talking to people about how they think that would work in their companies. And given the widespread lack of familiarity with healthcare
Yeah.
and the values and principles that inform healthcare, it's a problem.
Yeah, it goes both ways. It's harder to regulate and harder to defend on the other side too, right? It's a software, it's not a thing. Well, I think we've got 1 minute before we're due to finish up. So I'm going to start, just end with a final, just quick fire question. In your opinion at this point, who or what should governance data?
So could you repeat the question?
Who or perhaps what should be responsible for data governance in health systems?
So if I'll
Public
go first.
foundation that gets created specifically for that. Go ahead, Varun. Sorry.
No,
Go ahead.
that's pretty much, that was what I was going to say. It starts at that level, but I think that there has to be accountability measures that have to come from the federal level, I think.
Yeah, the federal, I mean, hopefully with this new legislation that passed in January, I mean, the federal government is the single biggest repository of healthcare data between CMS and the VA system. It's just rendering that data in a way that's intelligible to, big data analyses, and working through this issue of what we surrender when we engage with such federal systems. But this is more maybe like Glenn Cohen or Michelle Mello would be far better expert at this type of stuff than I would.
Great. Well, with that, we're right on the button at 4:55 ET. And thanks again to all our panelists. And I will pass back to Trey.
Thank you so much.
Thank you.
Thank you, guys.
Well, this has been quite an engaging day. I'd like to thank all of the speakers for great talks. I'd like to thank the session moderators for leading very engaging discussions, to the audience members for joining in and asking lots of great questions, and also to all the people behind the scenes who kept everything running real smoothly. So I think throughout the day, we really saw a broad range of applications of machine learning to genomics, ranging from basic science applications like protein structure prediction and elucidating regulatory relationships, to clinical applications such as looking at trying to diagnose genetic diseases in neonates. And we also heard about a very broad range of methods, including lots of neural net methods that have been adapted from image processing and language modeling, like convolutional neural networks and graph networks and transformers. And throughout all of this, I think there's a number of key recurring themes that we saw. And Trey is going to discuss a few of those in the next couple minutes.
Next 1 minute, yes. So Mark and I have been making notes all throughout the day. And just to highlight some of the key points we heard, starting with Eric Topol's keynote address this morning, how do we make the most of limited training data? You often have the case, especially in biology, where you have less data than you would like, especially as you move into the clinic. Eric and others mentioned the power of transfer, or so-called meta-learning, for leveraging a big amount of data you have in one domain, such as cell line models or mouse models even, to build systems that can then be more easily transferred to domains with less data, like clinical settings. The other weapon you have against limited training data is you can run synthetic data sets using generative models. And we heard about synthetic data from multiple speakers today. Brad Malin talked about how it can protect privacy by synthesizing data, especially in the early stages of a project. Eric talked about how it can sometimes lead to better accuracy. Sarah talked about how it's necessary, absolutely, for some population genetics applications. Of course, there are general pitfalls of using synthetic data. Namely, you want to make sure you're not learning more about your data generation algorithm than about real biology. but nonetheless, there was a very interesting discussion about the advantages of synthetic data. The other interesting theme that arose during the day was explainability and interpretability. Those are absolutely critical. I think almost everyone agreed, especially in life sciences, where you care typically more so about explainability or interpretability than you do about prediction accuracy. It engenders trust in clinical applications, especially. Christina Leslie showed us a beautiful example of Deep SHAP for how you can directly interpret her model to identify functional enhancers of gene expression, and so on and so forth. And then the other major theme, or another major theme raised by Pamela Sankar, was why progress in clinical machine learning has been slower than we'd like. And the major points there were, one, it's absolutely the highest stakes type of learning possible, so very different than someone's Uber failing to show up or failing to recognize your favorite cat on the internet. And so for that reason, we need to take things more slowly. And Malin talked about a sort of graded process for how we can do that. And so Mark, back to you for remaining points before we round this out.
Okay, I think we just have maybe 1 minute, but we also heard about a number of the ethical, legal, and social issues that have always been front and center at the NHGRI. So we heard about the fact that many populations tend to be underrepresented in genomic data sets, like GWAS data sets, which tend to be very heavily focused on people of European ancestry. And I think we might hear more about this tomorrow. We also heard about concerns with privacy preserving and technical ideas like secure multi-party communication that might help with being able to leverage data from different sites and more representative data without explicitly having to share it and reveal it. And we also heard some interesting comments from Brad Malin about the very real financial costs of trying to use a cloud. And in fact, tomorrow we'll have a whole session that's focused on resources for machine learning. So maybe that discussion we can postpone until tomorrow. I think we're right at 4:01, so we should probably wrap it up. But again, tomorrow there's another great set of talks and discussions, and we will start again at the same time tomorrow.
See you tomorrow, everybody.